computer. Right. So thank you so much for the invitation to be part of this, and I really appreciate your kindness and then your kind words earlier. And I really respect the idea behind the workshop and everything you've been doing concerning the center and also organizing this type of forum for people to gain insight into flies and everything. So just to a quick check. So am I clear enough? So like, can you hear me properly?
Yes, I can hear you.
Good. And does it say that I'm recording it as well?
Yes.
Very good. So yeah, so right. So let's start. So I'm going to introduce you guys to Drosophila genetics. So I'm just going to give a bit of introduction myself. So I started as a biochemist and then move into structural biology and then go into Drosophila. And then I have no knowledge about flies, and actually, so I learned the genetics like myself and through some mentors as well. So, and I would tell you that when I started, the resources are there, but some of them are not clear enough. Some of them you need to ask people, but there's a lot of resource out there. Is that okay? And one of the person that has really led the resource for Drosophila genetics, like that has really led that, is Andres Sprungkop. So I think you might know him. So Andres is in Manchester. He's a very, very nice guy. So Dr. Abolaji knows him very well. So he's really awesome. And he really did a very good job with the resource for Drosophila. And I can send you some of his resources later on if you need them. So, but yeah, so today I'm just gonna build on these things and also bringing together what other people have done and then tell you about what is it that we've got in Drosophila genetics, the tools, the techniques, and the magic of the fly. So, right, so the talk is going to focus on Drosophila, the genome of flies, how we determine sex in flies, how the naming techniques in fly, the development in flies, how we use fly to study things. So, and not only that, I'm also going to introduce you to some online resources and also tell you about Drosophila crosses as well. So beforehand, I'm going to tell you about what I'm doing personally. So now I'm really focusing on disease and drug. Personally, I'm looking at colorectal cancer for starting. I'm looking at other diseases as well. So, and the focus of my project is actually looking at not just flies, but also other systems to discover new drugs for patients. And this is a form of drug discovery that we're doing in the lab. Where is my... Right, so it's a form of discovery that we're doing in the lab, and it's just a beauty that, you know, flies are magical. They can do a lot of things. You can use them for a variety of things, but they're different from us. So the idea is that, can we use them as a tool for discovery and then combine them with other tools as well, and then that can allow us to build something better, if that makes sense. So and the idea of my own project is a form of drug evolution, combining fly with other resources like organ-on-chip system, and also computational chemistry and medicinal chemistry. So medicinal chemistry can be in form of natural chemicals, it can be in form of synthetic chemicals, it can be in form of other form of chemicals. And the beauty of it is that, you know, you can also start from, let's say, drugs are approved for a particular disease. So these are approved drugs for RAF. So these are RAF inhibitors, and they can be used. Some people use them for colorectal cancer and other form of cancer. And then you can begin to evolve this drug into better compound. Is that okay? So in fly, you can be like, okay, so this rescued the survival of flight, let's say 10%, but I can get something to about 80%. So that is the idea and the logic of the project I'm working on. And you know, this can take a bit of time. So basically you might have to synthesize, let's say, about 500 drugs or about 10,000 drugs to get to where you're going to. But you know, that's the challenges of beauty. So, and that's a beautiful thing. That's a beautiful thing in science. If it is easy, no one is going to do it because it's difficult. That's why we have to do it. So, and this won't be possible without this magical lab that I'm in. So I'm with fantastic people. Our lab is still young and growing in Glasgow, though old in the research field. And you can see my boss there. So Ross is really awesome. So, and I've got these lab groups and these teammates, I mean, and then they're awesome. also really awesome. So it's really good. So when you're trying to work in a particular, when you're trying to work on let's say a solution or a problem, so you have to find the right people to do the date with, or to go on a trip with. So it's really nice to be on a good journey with the right people. Right, so back to Drosophila genetics, tools and techniques. So I'm going to introduce you to some of the things in flies. One is the genome of the fly. So the fly has got about four chromosomes, not just about 4 chromosomes. And this is just the genome of the female. So the female has got XX, they've got 2 and chromosome 2, 3, and chromosome 4. And also the male has got, so the male has got XY, 2, 3, and 4, and these are really what provides what we call the genes. And just from our biology, that the genes are on the chromosome, and you've got the DNA and things like that, and then that leads to transcription, translation, and then you can get the protein. So these things are these type of arrangement is really on the Drosophila genome, and you can begin to pinpoint what is it that carries this particular protein phenotype or this gene phenotype, and then you can begin to be like, oh, this protein function is from this particular gene. So, and you can begin to also divide them into euchromatin and heterochromatin, and then begin to be like, okay, what are the functional parts, what are the non-functional parts, and then what is the size of the genome as well. And then you can begin to tell us how many genes are in this genome. So basically, it is powerful that we can begin to understand every part of the Drosophila genome at this point, because of advance in sequencing, and because we've really sequenced the whole genome of flies since about 21 years ago. So it's really nice to have an idea of what is it that you're working with. So that's the genome of the fly. It has got about 17,000 genes. So, and not just the beauty of the gene, but also genes that we don't know what they do. So the genes that are dead, but we don't really know what they do yet. So there's still tons of things to study and things to do. So, and also the genome of the fly allows us to be able to do something like cytological mapping, or looking at the position of genes. So let's say, for example, on 2R, which is the right-hand side of the chromosome 2, you can be like, okay, well, what are the genes that are there? Okay, I want to disrupt that gene, and then I want to see what's going to happen. Is it going to kill the fly? Is it going to do anything to their reproduction, to their feeding? So these are simple things you can do by just targeting a particular site on the genome, or in the genome of the fly. And also it allows you to do genetic manipulation. So that is just the beauty of flies. That is fly. So you can really play around with the genetics. So when I say genetics manipulation, is that, okay, you can be like, you know what, I want to add this carcinogen to this. What is that mutation that is happening? So you can begin to really lay the foundation of the particular mutation that you've caused. And then you can begin to look at, okay, how can I rescue this mutation? So looking at the genome of the fly allows you to do that easily and really in a simple and sophisticated way. So with all other organisms, especially organisms that are really used in the genetics world, it is difficult to do something as sophisticated as that, except aside from flies, actually, because, and that's why we work on them, and that's the beauty of working on them, because they can allow us to do a lot of really sophisticated things. Right, so I'm going to tell you about sex determination in fly, and actually how people have really worked this down, or how they've really walked through them as well. So sex determination in fly starts from something that is called the establishment promoter of the sex leader gene. So, and then that leads to splicing of that gene, and then you can get the protein, and then that gene itself leads to auto splicing of its own self. And then you can then splice other, and most of these things are really transcription factor, actually. So then you can splice them, and then that leads to production of genes that are specific to female. So it is an hierarchy of splicing events that leads to what we call a female animal. So, and also it is also a compensation between, do I have a very high number of X chromosome or not? So all these things goes back to the chromosome, and what are those things that are on the chromosome? Those are the genes. Is that okay? And it is what is going on with the genes that really leads to what we call, let's say, a female organism. And the same thing goes for the male as well. So the male lack that sexless establishment promoter, but they've got what we call the maintenance promoter, and the maintenance promoter doesn't do the same job as the establishment, and then you fail to have that normal splicing that is taking place in the female, and instead of having a female, then you're going to have a double sex that is for male, and then that leads to a transcriptional event of male-specific genes. Is that okay? So that leads to the formation of the testes and things like that. So and for female, that leads to formation of ovaries and things like that. So yeah, so those are the ways, or those are the pathways for sex determination in flies. And this is beautiful because we should be able to tell if we've got a male or female fly. And without that, really, that won't be really what we call Drosophila genetics, or we won't be able to multiply or really give rise to new things in the fly world. So we want to be able to tell whether we've got male or female. And I think part of the training as well is going to show you how to tell apart male and female, what things to look out for and things like that. So those are the number one, those are the first day training in the fly world. Right, so let's go to the nomenclature of flies. So that is where it gets interesting, because you really need to know what you're working with, and so the naming in flies or the nomenclature is really different from in flies to like let's say in mammalian system or in human actually. So there was some magic in the naming system, and it is really beautiful, and also sometimes can be difficult, sometimes it can be easy, but it's really nice to see how things were developed and what really brought about things. So the name of genes in flies. So let's say, for example, you're working with this particular gene and you want to give it a name. So let's say it's a gene that no one knows about in the past. So you can give it a name that you want. Is that okay? So, and you can be like, okay, the phenotype that I saw. So when we say phenotype, is that, okay, what you did was you fed this thing with the carcinogen, right? And then you saw that, okay, they were not moving properly. Is that okay? So let's say, okay, so they don't have any motion. So you can call them motionless. Is that okay? So then you can begin to figure out that what is that particular thing that is required for that motion. So then you're like, okay, it is this gene on chromosome 2R or on the 3rd chromosome. And then you can be like, okay, I'm going to name that thing motionless because they failed to move. Is that okay? So that is where the names come from. And for example, we've got white. So that is just because the phenotype lists, instead of the fly having a red eye, the phenotype led to white eye. So that was why it was called white. And there was this thing we call curly. So instead of having a straight wing, so they've got a curly wing, and we've got a transformer, which is really required, transforming from male to female. So basically the beauty of transformer is that, so you can do a cool experiment where you can actually have a male organism, but you can change, let's say, for example, the gut to female. So those are the, and then you can be like, okay, because, like even in human, the digestive system of male and female is different, right? So you can be like, why is it that some people feel, let's say some people are allergic to, let's say, for example, gluten, and some people are not. Some people are allergic to, let's say, oil, and some people are not. So, you can begin to ask that, okay, what if we feed this fly with this particular thing? What is going to happen? And it has got, let's say, a female gut, but it's still a male. So, these are crazy things that you can do with the fly. And those are really cool and really sophisticated things to do that you cannot do in other systems. And that's really beautiful. Right, so we've got so yeah, so another thing is that lowercase are for recessive phenotype. And then, so let's say, for example, the white that I mentioned earlier on is a recessive mutation, or it is a recessive phenotype. So you need to have, so for you to have a white-eyed animal, you need to have homozygous for white. So when I say homozygous, please, if you don't get that, just let me know. So homozygous for white, and then for you to get that phenotype. But for dominant ones, you don't really need to get homozygous. Heterozygous or hemizygous are okay for that, for you to see the phenotype. Is that okay? So, and then when we want to write the name, so you write recessive. For example, if I want to write white and I want to have a symbol for white, it's going to be W in the lowercase. But let's say, for example, dominant mutant, like let's say notch, for me to write notch, it's going to be capital N. Is that okay? So those are the symbols that we use in the Drosophila world as well. So, and then for protein, so because, and these are really important because when you're writing a paper and then you're writing about Drosophila, and because, you know, you want to publish, right? So, and then, you know, the people are going to look at, do you really understand what you're talking about? Do you really know what you're writing about? And then, so these are the simple things that we need to learn when we want to go into this type of system. So, yeah, so the proteins, you don't need to italicize them. And then you can start them with capital letter. Most of the time we start them with capital letter, but in those days people write them as an all capital letter. But you know, now in the mammalian world, actually even in human world, so like human genes, and then let's say mouse genes, I mean, so they go written in capital letter. Is that okay? And then but for Drosophila gene or Drosophila protein, we need to begin them with capital letters. Is that okay? That's what we do, rather than writing everything in the capital letter. But if you do that as well, it's not a problem. Right. So, and when you want to write the genotype of the fly, you have to write them according to the linkage order. So let's say, for example, I think I've got it then. One second. So let's say, for example, you want to. Right. So for the linkage, so you can actually. So let's say you want to write yellow and W. So yellow is a gene. Is that okay? And white is also a gene. Is that okay? So, but because yellow comes first on the first chromosome, you have to write Y first, not W. Is that okay? Which is also on the first chromosome. So you have to write them according to the linkage order. So assuming you write W first before Y, then you know that is wrong because that is not the way it's written in the fly world. Is that okay? So that's just an example for that. And also, so symbols of genes in different homologous chromosomes needs to be separated by solidus or dash. So let's say, for example, you want to write, so let's say you're on, so you want to write, let's say, something on the second chromosome or something on the first and second. So let's say we've got Y, and then we want to write, let's say, so Y and W is on the first chromosome, and then I'm going to come back to the next row that uses what I'm just using now. So, and then you want to write something on second. So let's say what is on your second is a particular gene that you've generated or something that you've really developed yourself. So let's say it's a UAS, let's say, for example, I'm just going to use MIC. So MIC is a gene. And then you can separate that from the homolog by, let's say, that is over what we'll call a balancer, and the balancer can be, let's say, curly. Is that okay? So you have to separate that, and then either you use dash, or you can use this. So you can use either of the two to separate your homologs. Is that okay? So that's another rule in writing your genotype. So right, so the next thing is that, so gene symbols on different chromosomes are separated by semicolon. So like I did already, so you can see that, you know, I separated that by semicolon. I should clean these, but it won't go away. So yeah, so I separated that by semicolon. And also, so that is just, so assuming you're on, so this is on the first chromosome, the YW is on the first, and then the MIC is on the second. So you have to separate them by that semicolon. Is that okay? So that's just another rule. And when you've got the absence of, when you don't have anything on the other side. So let's say, for example, when I wrote, so I'm just gonna write something else there. So here, let's say I write, sorry, that's good. So let's say I write, so that is Y over Y, right? So that is homozygous for that animal. And then I wrote, So and then we've got US make. Is that okay? So and then there is nothing. So I did not put anything below it or over anything. So that shows that US make is homozygous for that animal. Is that okay? And then assuming we go to the third chromosome, and then let's say we've got something like, then we've got let's say a balancer that is called TM6B. So we can never have a balancer that is homozygous. So that should be over something else. So then that would be MKRS, just for example. So we're going to come back to all these names later. So yeah, so that is something. So when we've got, let's say for example, that US make, and we don't have anything, is not over anything, is that okay? So that shows that is homozygous for the strain, or that is just homozygous. So when someone gives you that, or you requested that from somewhere, and then you see that, then you know that is homozygous. Is that okay? Right. So moving on. So right. So we've got that. You can generate your own stock. Is that okay? You can generate your own transgene, or you can also request from other people, right? So some people they've got their own mutation. So basically they did a bit of, let's say, genome-wide screen and then generate this mutation. So what they can then do is, okay, we found that we've got several mutations on the same gene. Is that okay? So for you to be able to identify which mutation is which, you have to separate them by something. Is that okay? So let's say, for example, like the white that I was mentioning earlier on, so assuming let's say you've got white 1, white 2, white 3. So you can be like, oh, white to the power of 1, white to the power of 2. So the same thing goes for these as well. So Rossi is another gene name, and then that also gives eye phenotype. Is that okay? A very dark eye phenotype. And you can be like, okay, Rossi 1 is the first mutation of that particular gene, and then Rossi 2 is the second, Rossi 3 is the third. So you can have a superscript for the type of mutation, and that gives you the identification number of that mutation. Is that okay? So that's another way to write your own, to actually write the gene or the mutation name or the allele name. Is that okay? Right. And when you're writing a particular genotype, or you're writing something, and for example, you're like, and actually I've given this illustration here. So you've got white plus. Is that okay? So white plus means it is wild type for white. Is that okay? And white minus means is a complete deletion of that. Is that okay? So it means you don't have, so that particular thing. So when you've got white plus, it means that particular thing that gives that phenotype is wild type is there. Is that okay? And when you've got minus, like, it means that you lack that. So for example, let's say you've got this particular gene. Let's say you've got a gene called scribble. Is that okay? So scribble minus means that you're lacking that gene, but if it is scribble plus, it just shows that it's wild type. So if you're referring to plus, you don't really need to write it. It's just like people will know that, okay, that is white plus. Is that okay? And white minus just means that, oh, that is lacking that particular gene. Does that make sense? So that is the way you write the genotype in Fly. So, and also you can describe the allele as well. So you can be like, okay, these things are temperature sensitive. I've got an allele that is sensitive to temperature. Let's say, for example, you can grow them up at 18 degrees, but they're gonna die at 25. Does that make sense? So, and let's say you fed this thing with, let's say, carcinogen or anything, or a chemical, and then that causes that particular phenotype. You can be like, okay, something is in that fly that leads to this temperature sensitivity. Is that okay? And also, let's say, for example, you're working with an enzyme. Is that okay? And you know that this enzyme has got an activity. Let's say is a glucose enzyme or glucose phosphatase enzyme. And then you can be like, okay, it has got this activity. So you can be like, oh, yeah, what can we do to remove the activity? So am I going to generate a mutation in these? And then you can test that against the substrate and be like, okay, no, it hasn't got this activity anymore. So those are the things. So you can describe your allele by this type of things. So you can be like, okay, it is null for this particular activity, or we mutated the catalytic site and then we don't see activity. Then you can be like, okay, it is null for the activity, but maybe still functional for some other things. Is that okay? Not necessarily you're going to lose the whole thing, but could be functioning, because you can have a mutation in a particular site and it doesn't disrupt the interaction of the particular gene or the protein with other, like if it interacts with other things. So the other arm of the protein could still be functional. And then you can just name that particular allele itself that, okay, this is not allele, but can still interact with other things. So, and then we've got like recessive, we've got dominant, right? So those are the things that those are the names that you can give flies. But these names, so except for the temperature sensitive one that people still use, most of these are not being used these days, really. So, and then you can have your transgene and transformant. Is that okay? So, and that is really the beauty of flies, and which we're going to get to later on. So, for example, you've got this particular transformant that someone sent to you. They can give you a full name of it. Is that okay? So the P element vector is just P. Is that okay? Yellow is the gene, is a gene. So let's say it's going to have a yellow body if it is homozygous, and then white plus is going to have, it's just like it has got a bit of a red eye. Is that okay? So because it is white plus, is that okay? And then you've got 10 times UAS GFP. So so this is just telling you that this is the full genotype. So this fly, it has got a P vector or a P element that carries a UAS, and that UAS element control the expressions of the expression of GFP. So basically, when you cross this to, let's say, for example, a GAL4 system or a GAL4 driver, then you can see GFP in the offspring. And then we're going to look at some examples of that later on. So, and also you can write this in two ways. So you can be like, oh, the cytological position of this insertion is 68A4 in the fly genome. So you can, so that's the beauty of working with fly, because you can really map out where is the location of everything in the genome of the organism. And also you can rewrite that and be like, okay, you know what, I'm going to write everything, and then I'm just going to put at P2. So at P2 just shows that it's on the third chromosome. So, and then we've got location, like I mentioned earlier, that there's a cytological location of everything on the genome. So, and this has been mapped out for every gene in the genome as well. So you can be like, okay, that is the location on the cytological position, and then that is also the location. It's just the same thing. Is that okay? And the way you write it as well is right. So you can use square bracket, you can use parentheses. So that is the way it's written. Is that okay? So, and sometimes we don't write all this in full. So not necessarily, so when you're trying to publish something, you don't really need to write all this in full. So you can be like, okay, so you can actually write USGFP and people will understand what you're saying. Or you can be like, okay, USGFP on TED. So people would know what you're saying. But it is very good for you to have that in your own probably lab notes, so that if someone try to be like, you know, probably you're publishing something that is fraud or something that is not true, then you can be like, you know, that is the location, that is what we found, and you know, you can show them. But in publication, USGFP on third or on second is more than enough. Right, and the last rule, and actually the biggest rule, is that, you know, never trust any stock. So you have to make sure that you verify the stocks before you start using them. So not necessarily that people do that intentionally, that, you know, probably they're going to destroy the stocks intentionally. No, but there could be mistakes. You know, these things have been from generation to generation. So you need, so for example, if someone tells you that, you know, this carries this particular gene, the first thing you can do is, okay, what is that gene? Okay, the gene is a kinase, and then it affects this particular substrate. So the first thing you can do is actually you can just PCR the kinase and be like, okay, can I see the gene? Is that okay? So you can cross this to a GAL4 that will allow you to really express the kinase, right? And then you can be like, okay, can I see that gene in the offspring? And then you can verify that, okay, yeah, it carries that. And also, if it is a mutation, you know, you can PCR it and send it for sequencing, and people will tell you that, okay, yeah, that is actually right. Is that okay? So those are the things things that you can do to verify. And also you can use antibody as well. So you can do Western on them and be like, okay, I can see binding of my antibody to this particular protein, and yeah, that is straight is there. So these are things you can do to verify the stock. And sometimes you can just do simple verification, because most of the stocks that we use these days, they are transformants. So most of them will carry what we call the P element. Is that okay? And then the P element is actually often tagged, or most of the time they will have like a white, like a red eye or yellow eye or orange eye. So you can tell that once your fly is probably losing the red eye or white eye, then definitely something is wrong with the stock. So please, you need to always check that. It's good to trust, but it's very good to always check. Right. So any questions so far? Any questions so far? Doctors? I can't see anyone.
Okay. I have a question.
Okay. Yeah, please do. Please do.
Yeah, the last point, the last before the second one, the transformers and transgenes.
Okay.
Yeah, I want to get, what does the, you wrote this in two ways. Can that be PYW?
Yeah.
What is that ATTP2?
Very good. Very, very good question. Yeah, so ATPP2 is just so we call them, like, it's just an ATP site. Is that okay? So there's ATP2, there are other ATPs that I actually have forgotten now. So there should, yeah, ATP2, yeah, I've forgotten the rest. So, but there are several of them. So basically, this ATP2 is just telling you that this is on the third chromosome, and it is just a site that this particular P element was inserted into. Is that okay? So I'm going to show you what, like, the way this is generated later on in few slides, in next few slides. So it is just that you've got your own fly. What is the location of this fly in the genome? And then you can begin to really do other things with it. You probably want to recombine it with something. So that is the reason why, one of the reasons why we really need to know the location of this thing is that, okay, do I want to recombine it to something else? And the ATP2 site is just the same thing as the 68A4 site.
Okay.
Does that make sense? So
yes,
so I wrote that in two ways, so that so you can write it this way. People will understand that, or you can actually go to the cytological position and be like, that is the position, and then write it that way. Does that make sense?
Thank you. Yes, thanks so much. Yes, sir. Thank you.
No problem at all. Right, someone raised their hand, but I can't because I closed my thing. So yeah, have we got any other question? You can just go ahead and ask, honestly.
Hello, are you hearing me?
I can hear you clearly.
Okay, please, there's a point there, allele identification often carry identification number. I don't get that place clearly. Can you please come as in recite that area? I'm trying to explain more better.
No problem. No problem at all. Yeah, so alleles identification often carry the identification number. That's correct. So what I'm referring to there is that, so let's say, for example, so in your lab, let's say you did what we'll call like a genetic screen. Is that okay? So one way to do a genetic screen is you can induce mutation just randomly in the genome. Is that okay? And then once you induce this mutation randomly, let's say, for example, then you're now saying, oh, there's a problem with the eye. Is that okay? Just for example, there's a problem with the eye of the fly. And then you realize that actually the position that causes this mutation is the same. So it is just a gene that it is a disruption in one gene that causes that mutation. Does that make sense? And then in that one gene, you've now got several mutations in the gene. So let's say the gene is about, so the size of the gene is, let's say, 400 base pairs, just for example. That's 1400 base pairs. Is that okay? And then let's say the protein, I don't want to go to protein, but let's say the protein size has got, let's say, 400 amino acids, and then the amino acid 1 to 1 to 200 has got 2 mutation. Is that okay? So there's one mutation that also gives the same phenotype as the rough, let's say rough eye. Is that okay? So, and we've got mutation from amino acid 1 to 200. Is that okay? So one of the mutations you can be like, okay, that is, let's say the protein is glycogen phosphorylase, just for example, glycogen phosphorylase. So you can be like, okay, glycogen phosphorylase 1 has got mutation as at site 1 to 200. Makes sense? And then from site 200 to, let's say, 300, you've got another mutation. So that can be glycogen phosphorylase 2. And then from 300 to 400, you've got another mutation. So that can be glycogen phosphorylase 3. Does that make sense? So because, you know, these are on the same protein, or because the gene codes for protein, right? So on the same gene, then you can use a superscript to name them 1, 2, 3. So that's just the identification number that you're giving to them, not necessarily in that same order, because in those days when people were doing this, there wasn't any form of sequencing to tell them that, okay, you know, that is the location of this particular amino acid, or that is the location of this disruption. But nowadays, that due to advance in sequencing, right, we can tell that, you know, it is actually mutation 135A that causes this. Does that make sense? So we can really go to the number rather than using the superscript. So we rarely use the superscript these days. Does that make sense? Actually, people go in detail to tell us what particular mutation is actually going on. Does that make sense?
Yeah, good.
Right, thank you.
Thank you. Right.
Right. Have you got any other question? Okay, so moving on. Yeah, please go on.
I still don't understand the transformants and transgene, especially the brackets, the parentheses. I still don't get. I still don't understand.
Oh, very good, very, very good point. So assuming, so let's say, for example, you go to, so Bloomington, I'm going to always refer to Bloomington because it's really a very vast resource and really, really, really a lot of work has gone into it. So Bloomington and Flybase, so they've got sections where, you know, you're going to see the name of the gene, you're going to see all these names there. Is that okay? So assuming you're saying something like this, so the P square bracket 10 times US MCD8 GFP, is that okay? It is the name of that particular strain. Is that okay? What this means? Okay. So actually the bracket, not really of a big deal. The bracket doesn't really change anything. Is that okay? Honestly, people go away with all the brackets and things like it doesn't really doesn't make any difference. But what really matters is what you're really looking at. Is that okay? So P stand for, that is a P element. So it is an insertion into the genome. Is that okay? It is not part of the genome. So flies doesn't have GFP. Does that make sense? So it is not part of the genome.
Okay.
So you inserted the P element into the genome. So that shows that, oh, you know what, I've inserted something. So then what you've inserted then carries yellow. So it can give the organism a yellow body. So normally flies are not yellow bodied. Is that okay? And then it can get, so because you insert this into a white-eyed fly. So I'm giving this white-eyed fly a red-eyed because I've inserted this P element.
Okay.
That makes sense. And that P element also contain 10 times. So basically, so when I say 10 times, it just shows that, you know, you've got, so this, I'm going to show you later on the vector that is used for this type of thing. So the plasmid. So in your plasmid, you've got 10 times sequence. So you've got UAS sequence in 10 repeats.
Okay.
And on that and next to that UAS sequence, you've got a membrane bound CD8 GFP. So CD8 is a protein, is a fusion protein. GFP is a protein. Is that okay? And you know, you've got the gene that codes for them, right? So you've got this on that in that vector, in that plasmid.
Okay.
And what have I done? I have inserted them into this cytological position, into ATP2 site or C68A4 site.
Okay.
Does that make sense? So, and you know, so that is just, so that is the name. So it's just a detailed name of, so now as in you give this to someone, so they will be able to tell. So they will be able to tell, okay, what phenotype should I look out for? If this is homozygous, they're going to be yellow body. They need to have red-eyed. And then when I express something, I should be able to see GFP. And then they are on the third chromosome. And this allows you to also, you can then track, because this is just a stock. You're going to play around a lot, that you have to set up some crosses, and then you need to be able to track them over time. Does that make sense? So it's not just about the stock, it's about, okay, I want to combine this with something else that we're going to show later on actually, and then just a bit of the background into the crosses. And then you can be like, okay, this is on the third chromosome. I can track it that, okay, I should inherit it on third. Then you can design your crosses, and then you can play around, and then you can do some experiments as well. So that is the key. That is the beauty of writing this in detail. And you know, like on Bloomington, on Flybase, they need to write in detail because you know it's for the world. So everyone is on it. Is that okay? So, but assuming you are in your lab, you can be like, you know, okay, I don't need to write this detail. What you just need to write is, you want to write UASGFP on TED. I know that my GFP is on TED because you also need that to track it. So assuming you only write USGFP, then you will be like, where, how am I going to track it? Where is the location in the genome? Does that make sense?
Yeah.
And also assuming you want to recombine this, so there's a recombination trick in Fly as well, that you know that your Fly is not carrying something already, but you want to combine that thing to it, and then you want to make sure that it carries it. So you need to play some recombination trick and then you need to be able to tell that this location, if I've got something in this location, it can recombine with another something in another location. But if you don't know the existing location already, you won't know whether it's going to work or not. But, and also actually you can calculate it mathematically. So that's another beauty of that type of experiment. So you can do that. You can be like, okay, this proportion of this organism is going to carry my recombination. So we can talk about that later on as well. Is that okay? So,
okay.
But these,
so is,
sorry. Okay. Continue.
So is ATTP2 also a location?
Exactly.
For the, okay.
Yeah. So at P2, at P40, so that's coming back here. So at P, we've got P40. So those are location. Those are just, so it's because, you know, 60, so okay, we've got 68A4, we've got 65B something. So we've got the cytological location. Is that okay? So those are the numbers, but not everyone will remember them. And, you know, people can still remember at P2, at P40, you know, people can still remember those ones. So that was why people use that. But the real, so, and that is just that's a location. It's just like a slang, or how would I put it? So, or for people to remember. So, but the real location of that is actually 68A4 on the genome. Is that okay? So,
all right. Thank you, sir.
No problem. Thank you so much for the question. Right. So are we okay to move on, right? Okay, cool. So, right, so I'm just gonna show you, so that is about naming, and please, if you've got other questions about that, and or you want me to go back to other things, just let me know. I'm happy to always move this slide around. Right, so I'm going to tell you about Drosophila and development. So, which is really one of the beautiful things about Fly, and I really want. So, if you've not registered for the Dros Africa workshop, so you should, because you're going to enjoy like those that really did a lot of discovery thing in fly, and actually really won Nobel Prize for that. So it is something that you should really go to, and it's going to tell you about the beauty of developmental biology as well. So Drosophila has been in the forefront of developmental biology, and also not just for developmental biology, but actually organism development. So we've got the embryo, and you can begin to look at every tissue in the embryo, and also you can begin to map out what they're going to develop into being. So this is just an example of the gut. So you can see from embryo, you've got the guts that develop into the larvae and into the pupae, and also leads to give rise to the adult gut. So, and it is beautiful that because you can then use all this for study. So you can be like, okay, I've got this particular gene. Is it going to affect the embryo gut? What is it going to do to it? Okay, if it doesn't affect the embryo gut, what does it do to the larvae gut? What does it do to the adult gut? And also the beauty of fly is that you can actually look at this particular stage individually. So you can be like, okay, I want to start looking at them at embryo, or I want to start looking at them at adults. So that is the beauty of the trick that we've got in the fly world. And also these are things that you can do in your lab as well once you get started. So so that I love these pictures. So I just love to show you. So that is it about development, and it's just about the life cycle as well. So takes about 10 days at 25 degrees or 10 to 11 days. So, and then I think that is the next slide. So, so flies can grow at variety of temperature. So, and actually that affects their development over time. So at 12 degrees, they can grow, like the life cycle takes about 50 days. And, at 16 degrees, about 25 days, at 18, and then at 30, about 6 days. So, but they've got limit of temperature where they can grow in. So, and I think there are some flies that probably resist temperature. I'm not very sure of that, but they cannot survive in probably minus 20 degrees, or they cannot survive in like 40, 45 degrees. Does that make sense? So there's a limit to temperature as well. So, but it's really beautiful that, we can rear them at variety of temperature. And I think, for example, in Nigeria, you've got this temperature range that actually you can put flies in your lab and, they will survive. So that's just the gist about that. And yeah, Andres, so I mentioned Andres earlier on. She has done a very good job on putting together a lot of things. And this is just showing you that, flies in human, we can use flies for discovery, several discovery, not all discovery, but, they've got a lot of things that they've got a digestive system, they've got the nervous system, and actually the nervous system has been flies have really pushed that forward a lot. So, and actually people have really used this, they've really developed a lot of tools in neuroscience in flies actually. So, which is really, really beautiful. And also the body plan as well. So the organization of our body, they've got organization as well. They've got circulatory and excretory system as well. So it's just showing you that, not just that you're using fly for some random stuff, but actually you can use it to tackle some difficult questions that are pertinent to human. Is that okay? Right, and also we can begin, so not just studying fly as a whole organism, but actually you can look at individual tissue in them. So you can be like, oh, I want to look at the eye, I want to look at the leg, I want to look at the wing. You can look at the eggs, which is something that I've worked on in the past. So, and you can look at the testes, you can look at different part of it, and you can begin to ask, okay, I've got this particular gene. Okay, I want the gene to probably give me some phenotype. What's the phenotype in the wing? What's the phenotype in the egg or in the testes? So you can begin to study this at a very, very low resolution rate. And, you can begin to look at individual cell actually in flies. So, which is really beautiful because not every system can give you that level of diversity. So, sorry. So yeah, so that is it. So you can study a lot of things. And the thing is, you need to be able to, not really difficult, but dissect out the tissue that you're interested in, and also probably have a microscope to look at it. So that is a bit, that can involve a bit of finance and something like that. But outside that, if you've got the microscope, you can do a lot of good things with the tissue as well, so which is really nice. Right, and in flies, the bread and butter of flies is that we've got balances. So without balancers or markers, then there was nothing like fly research. So fly research is still really standing on this type of balancers or markers. So for example, you've got for the color of the for the color of the body, actually you've got the wild type body, and then you've got the ebony body that is a bit darker, and then you've got the yellow body that is a bit lighter. So and then you can begin to tell this fly that we've got is yellow body. So why is it yellow body? What location is the yellow on? Then you can begin to figure that out, and then you can track that over generation as well. And then you've got the eye phenotype or the eye markers. So you can have a white-eyed fly. You can have the mini white, and then you can have the bar eye, which is really cool. You can have the drop eye. You can have the eve fly. So these are real markers. These are visible markers that you can see, that they're visible, that you can see. I mean, so you can really see them. So which is really, really nice, and play around with them under the microscope, and you just feel easy with happiness, I think. So, and then you've got the wing phenotype. We've got the curly wing, we've got the serrate wing, there is the vestigial wing, we've got serrate, So those are the main wing phenotype that we've got. Also, we've got bristle phenotype. So instead of the wild type has got just 2 bristle next to the eye, but humoral has got multiple, and pin has got a bit of a short one, and also stubble really, really short, and singe is a bit like squiggle one. So these are phenotypes that actually, they really helps us a lot. They really help us a lot in the fly world. And without them, like I said, there is nothing like the fly genetics or just fly genetics. So yes, so those are the balancers and dominant marker. So the balancers are very good because you can those can help you to maintain a stock. Is that okay? So dominant marker allows you to look at inheritance, but actually, not all dominant markers are not balancers, but some balancers have got dominant marker, if that makes sense. So for example, like curly. So curly serves as a marker because it has got a curly wing and also a balancer, and is on the second chromosome. Is that okay? So how do I know this? It's just because it's written in the textbook, and it's the fact, it's the reality. Is that okay? So you can see these things. That's the beauty. It's not abstract. Does that make sense? So it's the beauty that you can see it. Okay, this is scary. What location is on the second, and you can track that as well over the course of your crossing generation. And yellow is a marker, but it's not a balancer. Is that okay? So an animal is gonna have a yellow body, but it doesn't. Like it cannot serve as a balancer. So balancers allow you to really maintain your stock, but yellow doesn't allow you to maintain it. It's just there. Does that make sense? So those are differences between markers and balancers. And and when you check like online, for example, in Bloomington, you can tell when they're referring to like balancers or markers. Is that okay? And they're not, we don't have to, like, we rarely use, actually, so the number of balances that people use is about, probably routinely use about 10 of them, or actually 5 of them. Is that okay? So for the first chromosome, people use FM7. So that is really the key balancer. And like, you don't really, like, that is just general. So because it's first, so first multiple something. So that is FM7. Is that okay? And on the second, people use Kelly a lot as the balancer. Is that okay? And on third, people use MKRS or TM6B as a balancer. So those are the major balance. That's about 4 or 5. So those are the major balances that we use. But the rest can be markers. So and then you can begin to ask, or even you can ask that, what chromosome is this on? And people are going to tell you that, we've got this balance on so chromosome. Is that okay? And when you see something that is different from the wild type, you need to ask that, why is it like that? So you have to ask that question. Is that okay? So yeah, so that is it for balancer. Please, if you've got questions, you can just, you can poke me, and then I'm happy to stop and attend to them. Right. So I've shown you the markers and balances for adults, but the beauty is that we've got some for the larvae and the pupae as well. So we've got the wild type for larvae, so this is a larvae, and then we've got what we call the black cell. So this is just a dot of cell around the body, and you know that's actually a marker. And also we've got normal pupae for wild type, and then you've got what we call it tubby, so it's shorter one. So, and also you can have that as a larvae as well. So you can have a short larvae. So you can then begin to tell that, okay, I've got this particular gene on this chromosome, and so because the beauty is, assuming you've got a gene over tubby, and then you want to inherit that particular gene, so you can be like, okay, I will only look at the non-tubby because I know that tubby is the second. So you know these are homologs, right? Your gene is over tubby, so you can be like, okay, tubby is the one that like is going to be short, and the flies are going to inherit it, but my gene of interest is going to be wild type, so I can see. So you can then compare even within the same vial of organism that, okay, I've got the daughter and then the daughter and the sibling, then you can compare them. So between non-tubby and tubby. And also you can tell between male and female in pupae especially. So the male has got this genitalia here, and then the female lacks it. So you can actually track that from actually in larvae as well. You should be able to see this sex combo or something, so that is in the male and not in female. So yeah, so those are the balances, those are the markers. So, and you know, these are very important for Drosophila genetics, and really for if you want to work with flies, you really need to look at them and really be able to tell which is which, and which will allow you to actually track your inheritance as well. So if you want, so we can always go back to these slides for questions, please. So right, so yeah, I'm going to show you a bit of, yeah, Mr. Adeyemi, you've got a question. I think you raised your hand.
Thank you.
Right, thank you.
How do we maintain our stock using balancer? How? Can you explain? Can you share more light in a practical manner?
Very good. So I'm just going to switch to pen just to make it small. So I think we have a question. Oh, yeah. Okay, so I'm just gonna, so let's say for example,
you get me?
Yeah, I can hear you. I'm trying to answer the question. So let's say for example, you've got sorry. Are we good? Cool. So yeah, so let's say for example, so to maintain your stock, let's say you've got thinking. So let's say you've got something on the second chromosome. Is that okay? And that thing is, let's say it is you've got like you clone human gene into fly. So and the human gene is, let's say, an exokinase, just to make sure that a bit of like that is in human, right? So we've got exokinase, and the exokinase that you've got it on the second chromosome. Is that okay? So, and then you've got, let's say, white plus on the first, just for example, and then you've got your exokinase. So I'm just going to write X, and then you've got over curly, just for example. Is that okay? So this exokinase is not just exokinase. You can have it in various forms. One, you can, let's say for example, Drosophila has got exokinase as well. You can be like, it's a mutation in exokinase. Is that okay? Or you can have, let's say, UAS exokinase. So that is like, for example, you want to increase level, you want to overexpress exokinase in the system. Is that okay? So that is one thing. And or you can have, let's say, exokinase 1. So that's like a mutation. Is that okay? And one thing is that most mutations generally, I would say most of them, or even maybe 90% of them in fly, they are homozygous lethal. They are homozygous lethal. Is that okay? So assuming you've got X1 over X1, that's going to kill the animal. Is that okay? So, but you need a balancer so that you can always inherit, or you can always get back your X1. So let's say, so we're gonna go to crosses in very soon, and then you can begin to appreciate what crosses is gonna allow you to maintain your stock and which is not. So this X1 is never gonna have X1 over X1. Is that okay? That's one thing. And also you're never gonna have Kelly over Kelly. So because that's gonna die, the animal is gonna die. So how would you maintain it? You need X1 over Kelly. Exactly, that's the way to maintain it. So the UAS, yeah. Do you wanna? Yeah, you can interrupt, no problem. Yeah. Have you got Mr. Adeyemi? Have you got another question? Not there. The network, sorry.
I said you can continue, please.
Okay, good. So yeah, so now the USX over Kelly, I would say most P elements are always homozygous viable. Is that okay? Based on like those are really worked with in the past. So they're like homozygous viable. You can do some trick to them that would make them non-viable, but they are mostly homozygous viable. Is that okay? So over time, you can have your UAS X over UAS X. Does that make sense? So that is okay. That's not a problem. Is that okay? Because over time, because you know when you grow this up, Kelly over Kelly is gonna die, but US over US is happy. So over time, you're gonna lose your Kelly. Is that okay? So over time, you can end up with this. But for a mutant that will kill the animal, you need Kelly to balance to really maintain that, to balance that. Is that okay? So that is why they'll call it balancer, so to balance the system. Is that okay? So, and without that, then you won't have anything, because then, you know, once you've got X1 over X1, then everything die, and then the stock is gone. But because you've got X1 over Kelly, then you're happy. Does that make sense a bit?
Good.
Right, thank you. Yeah, so please, if you've got other questions, let us definitely address them. So right, so moving on. So I'm gonna talk to you about Drosophila, some technology in flies, and actually brush through this a bit. Yeah, I'm seeing your hand up, Mr. Adem. Have you got another question? Or is it just the computer that is messing with us?
Good. No.
Oh, pardon me. Okay, right. So there was this thing that we call the classical forward genetic screen, and I think I've really alluded to that a bit earlier on. So this is really looking at, so what you can do is just what you're doing in the lab, feeding things with carcinogen. You can feed them with mutagen as well, and then you can begin to look at, okay, what mutation is going on? Is that okay? And then you can have some stock of various mutation. Does that make sense? So that's the type of thing you can do, and there's a way to do that. So basically what you do is like I've said, you feed the male especially, and then you cross them to another female, and then you can pick out a male from that cross. So you cross these to these, pick male from that cross, then you cross them to another set of female. Is that okay? And then you can look out for phenotypes. So let's say, for example, you're looking for a ring defective phenotype. You can be like, okay, all those that have got this type of ring, they are the same family. All those that have got different type of ring, they are the same family. So, and then you can test the various way to do this. You can begin to probably sequence them to look at the type of mutation that is actually going on. You can cross them to, let's say, other strains to look out for what is that site that is actually leading to that mutation. Does that make sense? And this has been done like 50 years ago, 40 years ago. People do this a lot, but these days, it has been replaced by, you can do RNA screen. So the RNAi screen, sorry, or you can do CRISPR screen. So instead of feeding things with mutagen, is that okay? You can easily actually go targeted. So for example, you want to look at all the kinases in the Drosophila genome. You can ask, what will happen if we knock down all these kinases? Is that okay? So assuming you want to look at all the enzymes in the glycolysis pathway, so you can be like, okay, I want to knock down all these and then see what happens. And CRISPR can allow you to really knock them out. So you can be like, okay, when I knock them out, when I remove them completely, so knockdown is going to reduce the level, knockout is going to remove them completely. So when I remove them completely, what will happen to this fly? What will happen to the glycolysis pathway? What will happen to those that are upstream? What will happen to the Krebs cycle and things like that? So you can begin to ask that type of question, and not just for pathways, but also for signaling pathways as well. Not just for metabolism pathway, but also for all the signaling pathway. So you can begin to look at, let's say, the RAS pathway and ask question about what will happen when I knock down or knock out this particular family or this particular member of this pathway. So the classical genetic screen was really fabulous, and it was really nice in really identifying key stuff in flies. And actually, a lot of people did this and also won some people Nobel Prize. So it was really, really nice to do that. So, but like I said, there's a lot of advancement now, and we've really moved into the world of transformation combined with P element. So, and actually that is just a definition of Drosophila transgenesis. So you don't really pick a fly and then develop and then try and generate some mutation out of it anymore, but you can actually design some new flies and really target your question to those flies that you design. So, and that is the beauty of Drosophila transgenesis. So Drosophila transgenesis is just you taking a particular gene. So for example, sequencing has really helped us a lot. So we know that, okay, assuming you've got so let's say you've got insulin. Is that okay? Insulin. So you can tell the sequence of insulin. Is that okay? You can tell the number of amino acids. You can tell if you mutate this thing, what's going to happen. So you can pick insulin. Is that okay? And then you can be like, okay, what will happen if we clone insulin and then drop it into fly? So you can ask that question. Is that okay? You can be like, okay, what will happen if we like clone something else? It could be an hormone. It could because fly don't have an hormone. You can do something crazy like that. So like the hormone in fly is different from us anyways. So or you could also look at signaling pathways. So let's say, for example, you're interested in pathways that probably leads to hypertension, just for example. Is that okay? And then you know that, oh, this particular gene is been linked to hypertension. So let's say increase in level of the gene has been linked to hypertension. So you can do two things. You can ask, do we have like autolog of these genes in fly? That's the first thing that you need to ask. And then if we don't, then you can be like, okay, what if I clone this human gene into fly? What will happen? Is that okay? That's the next thing you can ask. So, and then to do that, you really need to synthesize this gene that you're interested in. You need to clone it. So this is a bit of molecular biology. Clone it, is that okay, into a plasmid vector that we refer to as P element. Is that okay? And then this plasmid vector will have several markers. It's going to have the white plus. It's going to have the antibiotics resistance. It's going to have the restriction enzyme site. Also, although this has got like Z, some of them will have like UAS. Some of them will have like, can be tagged with, let's say, MIC. You can tag them with GFP. You can do a lot of crazy, molecular biology is crazy, right? So you can play around with the engineering and do a lot of things. So you can then pick that particular vector that you've developed, so you can develop something really crazy. And like that is the beauty of fly. So you can do a lot of crazy things and then drop them into fly. So, but the beauty is that you can then tell the location of where you've dropped them. Is that okay? So that is really what the Drosophila transgenesis has allowed us to do. So you can be like, okay, I want to drop it in this location in 2 hours. So that is So 2R is just the right hand side of the second chromosome, and 47F is the cytological location on that particular chromosome. Does that make sense? So you can drop it in there and then ask the question. So no matter the location of where this thing is, most of the time they're going to serve the same purpose, but sometimes there can be, there can be variation. So that is one thing you can do. And this vector can be anything. Like I said, you can design anything. So although you need to make sure that, you can express can you express it in bacteria or something like that? And then you can then be like, oh, can I express it in fly? So you can do a lot of things with that. So another question you can ask is, okay, you can also synthesize things that will allow you to knock down genes. Is that okay? So you can synthesize like inverted repeats of a particular gene that target this particular gene, and then you can clone it. You can clone it, drop it into flies, and then ask, okay, when I express this particular thing under US, so this hasn't got US, but under US construct, can I knock down this particular gene? And then you can do probably Western, you can image them to see if you actually knock down. So so molecular biology has really helped Drosophila Transgenesis a lot, and people are doing a lot of magical things with them. So once you clone your plasmid of interest, you're happy, you make sure that all the sequences are in the right orientation, you're happy that, okay, I can get things going, you've got all your tag in, you've got all your restriction enzyme site in, you're happy with it. Then the next thing is, sorry. So the next thing is, then you need to inject your embryo. So embryo are going to be injected at a stage known as the syncytial blastoderm stage. And then you can inject just the P element, or you can inject something that is also called the helper. So the helper constructs allow for transgenesis. So transgenesis is that just similar to bacterial transformation. So you're gonna like open up the bacteria so that you can put something inside it. So the same thing goes for flies as well. So and then the bacteria is gonna incorporate that into the system. So flies can also, the embryo of flies can also do that. So then it incorporates that into the system, and the background of that system is white minus. So they are white-eyed. Is that okay? And then, so this is just telling you what the helper T cell does. And then at the end of the day, the helper element is not going to be inherited, because they're not going to be, they're not going to be, they don't have, for example, the ATP site or the site that will allow them to go into the genome. Does that make sense? They just help in the transgenesis. So then what you can then, so you inject this into the white eye, and then you cross these to other things, or you wait a bit. And so because you inject this into a white eye, cross this to another white eye, and then you can generate your transgene. So those that remember that this particular embryo is gonna give rise to adult, right? So some of the adults are gonna have white-eyed, some of them are gonna have red-eyed. So she's about right.
Sorry, I have sorry to interrupt.
No
now
problem.
the C there.
Yep.
You mentioned something. You said the injection of the P element into the larva. So that means we are inserting it into the larva, not the adult fly.
No, yeah. So actually, it is the embryo. So these are embryos. See that? So these are not
the embryo. Okay.
Exactly.
Okay.
So
we allow them to.
Okay. Yeah. Okay. So they're really, really small actually. So you insert them into the embryo, and then you allow them to develop into adult.
Adults. Okay.
Exactly.
I was thinking we are going to the insertion is going to be in the adult fly.
No.
So we allow them. So that means there's tendency that we may lose some, we get some.
Actually, people lose a lot. Yeah, so people lose a lot. So because that's so like assuming it's easy, a lot of labs will do this every now and then, but the probability of getting it right is really low. That's why you have to source it out to people. So people do this for us most of the time. Is that okay?
So this is what Bloomington does most times, and we collect from them.
Exactly, yeah. So Bloomington does this, like Cambridge, people in Cambridge do this as well. So Andrews was telling me that Manchester is gonna start doing this as well.
Oh.
So yeah, so like, and also like there are companies that are also like really companies that do this, but you have to pay them. So it's not done for free. But if you've got the technique, like if you've got the technique running in your lab, it's really good. It's very nice.
Now
So
I'm looking up that Dr. Abolaji will have that in next. Let me say in a couple of years we should have that.
Very soon. So yeah, but yeah, because it involves very good hand as well, because you're injecting this into really small, and actually it is called micro injection. So you're injecting this into very small organism or into very small.
So that means we are using those micro needles, or
exactly, yeah,
okay.
Micro needles, yeah. So and then you have to look at them under the microscope and then make sure that you're injecting the right thing. Is that okay?
Yes, thanks very much.
No problem at all. So yeah, so you're gonna cross this to another white hide, and then if you've got, it's just if there's remember that you're injecting the whole organism, right? Some of the chromosome will carry what you like, some of the cell will carry what you're interested in, some of them won't. So then you can begin to tell, okay, do we have our insertion of interest by looking at red hide? So remember here we've got white plus, so that will give the animal mostly a bit of a red eye, but sometimes it can be yellow, sometimes it can be orange. So, but at least you're gonna be able to tell apart white. Is that okay? So, and then you can pick those that have got your white, we call them white mini gene or your transgene, and then you can cross them and make a stock out of them. And not only that, people also like from the companies, people also balance them. So you can balance them over Kelly, over TM3, over SM6A. So you can balance them, and then assuming you need that, you can use that for other crosses as well. So that is how things are generated. So you generate, so basically you start from molecular biology and then you go into Drosophila. Is that okay? So that is how we generate transgenes.
Yeah, sorry, doctor. So now you mentioned balancing them. So you balance them based on the chromosome. If it's the first one, you use it, or if it's the second one, you said we use the FM7 for first one. Then, so that means if I see anyone that have the FM7, I should think that the gene of interest is at the first chromosome. Is that it? Okay, but if I have the third one, that is the MKRS, that will be the third chromosome. If I see that balancer, I should be looking at that my gene of interest is in the third chromosome.
Very good point. Yeah, so that is correct to a large extent, but sometimes, let's say for example, your gene of interest might be on the second, but people in the lab, they've got this, maybe they're using this for crosses. Is that okay? So they also have some things on the third. So some people can have balancer on just randomly on the third, and then, but you need to ask them that, what is the full genotype of this stock? So that can tell you where to look at. Does that make sense?
Yes.
But from this particular transgene, because it is only one insertion, does that make sense? So mostly your balancer is good, like I said, where you're looking at is going to carry your balancer, or your balancer is going to carry where you're looking at. So does that make sense? So if it has got FM7 balancer, then it shows that your stuff is on this first. And if it has got, like, let's say Kelly, it shows that it's on the second. And like you said, MKRS, it shows that it's on the third. Does that make sense? But
yes, thanks so much.
Yeah, but from other labs, you need to ask them that, please, where is the location, and they can tell you.
Okay, thanks so much, sir.
Right, no problem. Thank you so much for the question. Those are really, really awesome. Right, so where are we? Right, so I'm just going to show you some of the techniques now. So we've got the GAL4 UAS system, which is really, really awesome. And, that's what we use a lot in the fly world, and you know, it's really bread and butter of the fly world. And assuming you want to start something in your lab, so the first two things you need is you just need a GAL4. So GAL4 is gonna, so what GAL4 does is, so I just want to look at the way, so it is a transcription factor that allows you to, you know, binds to this thing, to the US promoter, and then allows you to drive genes, right? So, but for example, you want to drive something into the eye, like in the eye of the fly. So that was why it says it is a, so you've got a promoter that is really tissue specific or even cell specific. Is that okay? That you're going to combine with the GAL4. So let's say, for example, to the eye, you're going to have what we call the eye. It could be a particular gene that is specific to the eye. For people use a lot, the eye or GMR. Is that okay? And then that can be iGAL4 or GMR-GAL4. Is that okay? So that is a driver that can drive things to the eye specifically. Is that okay? And then when you combine that, then you need to cross that to what we call a UAS construct. So UAS, so that's another fly. Is that okay? And your UAS can carry GFP. Is that okay? So, and actually the beauty of that is, you know, when we're talking about checking your stock. So the first thing you can do is, you know, cross this part. Oh, someone gave you this GAL4. You know, it is expressed in the ovaries. Oh, it is expressed in the wing. Okay, you know what? The first thing I'm going to do is just cross that to GFP and see where the expression is. Because you can see GFP under the microscope. And for unlucky cases, you realize that you don't see GFP, you have to just bin that, or your GFP is wrong. So, you know, so those are the cool things that you can easily do, and you know, you can be like, oh, we found the right thing. Does that make sense? So that is one way to check your stock. So you cross that to UAS GFP, or you can cross that to, let's say you want to knock down a particular gene, you cross it to that gene that you're interested in, and then or to the inverted repeat that target the gene that you're interested in. So, and then you can knock down the gene. So knockdown constructs, you can get them from Bloomington 1 or VDRC. So these are two major places that you can get the knockdown constructs. Does that make sense? So, and you can ask question, what will happen if I knock down this particular gene in the wing? What will happen if I amplify, if I increase the level of this gene in the wing? So, you know, those simple questions, and, you know, becomes really sophisticated at the end. So those are the things we can do with the GAL4 US system. And then, you know, then you generate an offspring that carries both of them. Is that okay? So I've still got some crosses at the end that we can quickly go through as well. So that is actually what the GAL4 US system is all about. And we've got other systems that are also binary system, including the LEX-A, LEX-OP system. Use the same idea, the same methodology. It's just that the form of expression is different. And we've got the QF and the QUS system as well. So that is also the same ideology, but the transcription factor and the US element is different. So, but the beauty of these two other system, or the binary system, is actually, what the cool thing you can then do is, okay, I want to knock down this particular gene in the wing, but I want to increase another gene in the gut. What will happen to the fly? And then you can, you know, this that you're expressing in the wing is independent of what you're expressing in the gut. Does that make sense? So that is a cool thing about this type of tools on that independent. And, you know, you can begin to ask, is there any, we know about organ communication, or even tissue to tissue communication. So you can begin to ask, is there a communication between the eye and the gut, between the wing and the ovaries and things like that? So you can begin to ask those type of cool questions, and it's really, really magical and really beautiful. Right, so then we've got the Frt and Flp system. So this system is mainly used for clonal analysis. So what this does is that you can generate, so instead of you targeting the whole wing, compared like using the GAL4 US system, which you can also target some cell in the wing, but actually you can target clones. So you can target, like, let's say, assuming you've got a total of 1,000 cells in the wing, so you can target, I want to target just 50 cells. So you can target these things using what we call a clonal analysis. And the idea is really, you've got this, what we call the Frt site, that is targeted by the flipase. So flipase is an enzyme, is that okay? And then that flip out the Frt site, and then you can generate, so this is a bit of a random thing. So then you can generate random patch of clones, for example, in the wing, if you use a wing flipase. So assuming you use, let's say, for example, actin flipase, or a flipase that is expressed like in almost all cells of the organism, then you can generate, you know, patch of clones in the whole organism, but in every cell. Does that make sense? And also the beauty is you can then you can compare, okay, this clone compared to the neighboring cell, what is going on with them? And then you can begin to study, and this is the beauty of, this is what people that study apoptosis and you know, like they use this type of things a lot, because you can begin that, okay, what happened to those cells that are dying? Are they extruding? Is the other cell proliferating? So these are questions you can ask with this type of tool and technique. And also you can look, so you can use this for both, you can use it for US construct as well. So, but there's a flip out system that you can use for that, or you can use this for a particular mutation. So let's say you've got a mutation in let's say EPO. EPO is a protein that is part of a pathway that is required for proliferation. So, and EPO is a pathway that is also required for cancer. So you can generate a mutation in EPO, in the EPO gene. Is that okay? And then you can look at it in the wing and be like, okay, because EPO is required for proliferation, so when you reduce EPO level, cells should proliferate more. So then you can begin to ask that, okay, removal of EPO, what will happen to those cells? Is that increasing? Are they gonna have increase in size? Are they proliferating? Are they dying? What's happened to the cell? What happens to the cell in the neighboring environment? And you can ask those questions. So, and you know, you can stain for things as well. And the beauty is you've got this internal control. So because under microscope, you know, there can be problem with the laser and everything. So, but you've got this internal control that you can compare your mutation of interest with the wild type environment. It's okay. So that's the beauty. So every system has got its own beauty. Is that okay? So that's the beauty of the clonal analysis. So, this is just an explanation of that. So, and how the system work. And so assuming you've got this Frt site and then you've got GFP, so you've got your flipase, so you can flip out things, so you can remove things, and then that leads to recombination. And after recombination, then there's going to be mitosis, and then after cell division, then you can have some cells that are carrying GFP, and then you've got some cells that are carrying your mutation of interest, and then you can compare and look at them. Makes sense. So right. So another tool that I really love and really fascinated about is the Markham technology. So this is just a bit similar to the clonal analysis, but actually this is in combination with the Gal4US system. So so for example, you've got a fly that carries a mutation that you're interested in, and then you want to look at, okay, what will happen if I've got because as I mentioned earlier on, most mutation in flies, when you've got them as homozygous, the organism is going to die. Is that okay? So what you can then do is, okay, what will happen if I've got this mutation as homozygous in some particular cell type in the organism? Does that make sense? So, and then you can be like, okay, I want to look at the ovaries. So, and then you can ask, what will happen if in the ovaries, I've got some cell that are lacking a protein known as non-stop, and then some that have got this protein, what will happen to the ovaries, and what will happen if the population of this cell increase? So you can also compare population. Okay, when 3 cells are lacking this particular protein, this happen. When 10 cells are lacking, this happen. So you can also do that. And like I said, this is in combination with the Gal4US system, because you can label previously your cell in the clonal analysis, your cell lacked GFP, but in these actually your cell are expressing GFP while the wild type cell are lacking GFP. And it's just, so it's similar to the Gal4US system, because what happens here is that you've got the Frt and the FLP. And so once it shock the organism, the FLP, so let's look at this, actually this offspring. So you cross this to this. This is called a tester strain. I don't want to bog you with a lot of the detail. And then this is the mutation. Is that okay? So then you've got this offspring. Is that okay? Then the flipase is gonna do what we call the flipping out, flip out the FRT, and then there will be recombination. So because the FRT in this, so once you flip it out, then this is recombination. Is that okay? So, and then after recombination, you're gonna express GFP under the GAL4 system. Is that okay? And then you've got the GAL80 in the system as well. So GAL80 inhibits, so is an inhibitor of the GAL4 system or of GAL4. Is that okay? So cells that are expressed, so because you're recombining and flipping, right? So cells that have got GAL80 lack GFP. But cells that lack GAL80 have got GFP. So just here, when you look at cells that are lacking GAL80, so these are cells. So those are lacking GAL80, they've got GFP, and cells that have got GAL80, they're lacking GFP. Is that okay? Because GAL80 inhibits the activity of GAL4. And once the activity of GAL4 is inhibited, GFP is not going to be expressed. Does that make sense? So that is how the system works. But, and like I said, you can use this for several things. It depends on the type of question or answer that you're looking for. Right, so the final tool that I'm going to tell you about is a recent one and really, really powerful. So it's known as a CREMIC technology. And this is just another form of transgenesis and insertion into the genome of the fly. So basically you insert some construct. Is that okay? And mainly these constructs can be GAL4. Is that okay? And then what you actually do is you disrupt the gene in the system. Is that okay? So you insert it so that you can disrupt the gene. So these are also inserted randomly. So but with sequencing and everything, you can tell the location. So, and then you can look out for phenotype as well, that okay, if we insert this at a particular location, what will happen to the gene that is at that location? Because we know where these genes are. We've sequenced them, we know everything. Does that make sense? So then you can be like, does that disrupt the activity of the gene? So that is one thing that you can do with this particular system. And not only that, you can also flip that out. So you can flip out what you've inserted. So that's a power. So with the same fly, so this is going to give you a fly. So that's the starting point. So, and then with the same fly, and then you can be like, okay, that fly leads to this particular phenotype. So rough eye phenotype. So what if we flip out this T2A GAL4 that leads to that, then what will happen? Does that make sense? So, and then you see that everything is normal. It shows that what you've disrupted is the cause. Does that make sense? And then the next thing you can also do is you can also exchange. So you can be like, okay, I don't want GAL4 in my system, but probably you want GFP. So you can exchange this with other things as well. So that is another tool. So the paper is there. That's another tool. And also you can also rescue your phenotype. So let's say in your gene of interest, you've disrupted, let's say Yorkie. So Yorkie is another component of the EPO pathway. So you've disrupted Yorkie, and then you've seen the phenotype. So you can be like, what if I put back Yorkie in the system? What will happen? So, and actually you can express Yorkie under the control of the GAL4 that you previously inserted. So this is an insertion, is a plasmid. So like I said, insertion involves using plasmid, right? So you insert it into the fly, and then you can do all this magic with the construct, and which is really, really cool and really fascinating. And I think that is I've never seen any other technology after this ever. Like, so this is one of the latest technology in the fly world. So, and it's powerful, and you can give it a go if you want, and we can always talk about how to use it as well. Right. So this is the last part of the talk, and then we can go over several questions, and we can always revisit other things. So right. So I'm just going to show you some like a bit of background into how we design crosses in flies and how we use these crosses to like probably answer or ask some new question. So because that is really what we do. So we need to design crosses. We need to move around genes in the genome and things like that. So that's really the power of flies, and we need to do them because that's what research is all about, and we need to use that to tackle question as well. So so this is actually looking at a particular mutation. So a particular mutation in a protein known as ataxin 7. So ataxin 7 is required for several things, including it has been shown in cancer to be required for the thyroid cancer and incorporate with RAS. So, and then we've shown that it is required for proliferation in Drosophila egg as well. So it's a gene that I've personally worked on in the past. So, but not very common. And also actually, yeah, it's required for ataxia. So this is a form of neurodegenerative disease as well. So yeah, so, but this is, so you can pick anything. So we can go through your gene of interest and, you can pick anything. So, but the question is actually, it's just giving you a bit of a background on how to do some basic crosses and how to set up some crosses, right? So the question, the preamble to that is that, okay, you keep a fly stock that carries a homozygous lethal recessive ataxin mutant. Is that okay? So this is homozygous lethal because if you've got ataxin 7 over ataxin 7, it's going to kill the organism. Does that make sense? And it is wild type for the white locus. So basically it is wild type because it has got red eye. Does that make sense? So, and however, you want to do a recombination. So like I mentioned earlier on, you want to know the location of things to do recombination. And why would you do recombination with, for example, ataxin 7 is, for example, you want to look at if something rescued the phenotype of ataxin 7, and that particular thing is also on the second chromosome. Does that make sense? If it is on the third, it's easy because you can do some crosses to do that. But if it is on the second, you want to do recombination with ataxin 7 to see if you can rescue the phenotype of ataxin 7. Does that make sense? So that is one thing that this is useful for. So, but you cannot do recombination. I wouldn't advise you to do recombination in a red-eyed background. So because you won't be able to see what you're recombining, but you can do this in a white-eyed background. Does that make sense? And the reason for that is because when you're recombining to something else, just what you're combining to most of the time will have probably a yellow eye or orange eye or red eye. So you can tell that, I have done my recombination correctly. Does that make sense? So that is one thing. That is one way, or that's one reason why you don't want your ataxin 7 to have a red-eyed, to have red-eyed, I mean. So right, so following on that, what you want to then do is you want to write the genotype of the stock that you're interested in. So what stock? So we know that this is not the stock that we want. And then you want to design a cross to do that. Is that okay? So, so I think I've got this empty slide. So yeah, so let me so we've got ataxin 7. So if you want to take a snapshot of this, just to remember what the stock looks like, and then we can go through the genetics later, like in the next slide. So we've got ataxin 7. Right, so good. So right, so we've got we've got this stock. Actually, I need pen. Right. So our ataxin 7, our stock number 1 is semicolon to separate chromosome. So I'm just gonna do that. So remember that our third chromosome is wild type, so we don't really need to write that. So, but it doesn't matter. Is that okay? And then our second stock is, so we've got white-eyed balancer stock. Is that okay? So this is the beauty of balancer, because you can see it, and so not just balancer but also marker, you can see them, and then you can use them for your crosses. So you can use them to track your crosses. Makes sense. So yeah, so those are the two stuff that we want. And the first question says, what is the final thing that you're interested in? So your first question, so what you are interested in is because you want this to be in a white-eyed background. Is that okay? So you want your ataxin 7 to be in a white-eyed background. So the first thing you know that you need is, on your first chromosome, you want W over W, which is white. Is that okay? And then on your second, because you still need ataxin 7, you need ataxin 7. Is that okay? And then, because we know that ataxin 7 is homozygous lethal, it's going to kill the animal. So we cannot have ataxin 7 over ataxin 7. So we need a balancer. So our balancer is Kelly. Because you were only given these two, so you've only got these two stocks and you want to make something out of it. So you have to think of how to, have to move things around to get what you want. So Kelly is the balancer that we're going to use here. Makes sense. So you can stop me if you've got a question along the line, please. So right, so balancer so Kelly is a balancer that you're interested in. And like I said, we've got plus here and plus there. So let's keep the plus for now. So because we don't need them, so they're just wild type. So that's okay, we are happy. So now the next thing is the cross that you're interested in. So B, so your cross is going to, right, so the first thing you want to do with the cross is you want to think of, okay, where should I start from? So you know your end point, is that okay? But where is the starting point? So the first thing you need to do is you want to dilute this plus. That is the first thing you want to do, is that okay? And then you want to make sure that you've got W over W. So remember that this is like a symbol for female, and the male is going to be W over Y. Makes sense? And for the other one, that is going to be plus over Y. That's going to be the male. Makes sense, right? So you know that you can dilute this. So your first goal is actually to bring this to plus over Y. Is that okay? And to still keep this as W over W, or bring this to W over Y, or bring this to W over Y. Makes sense. So, but you want to make sure that you dilate this out. That's the goal. Is that okay? So W over W or W over Y, that's for male, plus for female, plus over Y. So the first goal is bring W over Y. So that will give you a male. Is that okay? And then what will happen to your second chromosome? You don't know yet, right? So, but the first cross you want to set up is you want to use female from these guys. So we're going to use, and then remember in flies, we always use virgin female. So is that okay? So then you want to cross that to male from here. So because that's female, over Kelly. And now you need to, so I can write out all the possibilities. So your possibility is, or your possibilities are, you can you can have W over plus. Is that okay? If over Kelly. Or you can have W over Y if over ataxin. Is that okay? Or you can have W over plus Kelly over Kelly. So what other possibilities So, good. So yeah, so W over Y. So we can have this. So because W is gonna, so when you're crossing, you're splitting just to the law of segregation and independent assortment, right? So you're gonna segregate the chromosome and then you're gonna inherit them individually. So you're gonna have, so W is like homolog of the homologous chromosome. So split that, you inherit that with plus because that's gonna split from Y as well. So that will give you W over plus. Does that make sense? And then also for the second chromosome, so you do this for every chromosome because every chromosome is independent of the other. Does that make sense? So then you need to split if, and you can inherit this curly with if. Does that make sense? Or you can have W and inherit Y because that is male. And then you can have your if and inherit ataxin. Does that make sense? Or you can have your W and inherit plus, and then you can have your curly and the other curly. You can have your W and inherit Y, and then you can have your curly over curly. Makes sense? But the first thing and the beauty of balancer is that those will die. You can never have them because they've got curly over curly. But these will survive. Is that okay? And what we are actually interested in is we're interested in this male. Makes sense? So that is how you, yeah, Olusegun, please.
Please, I just need a clarification.
Yeah.
What is it not possible that we also have CYO and ataxin 7 from this course?
Yeah, actually, that's a very good point. Yeah, so we will. So I will thank you for bringing that up. Good. So yeah, so we will also have, very good point. So we will also have W over W over plus atax 7. That's a very, very good point. And then the last one is W over Y over, okay, yeah, that's correct. Very good. So I'm happy that someone, yeah, makes sense.
So can we have ataxin 7 over if is it?
Yes, so that is still the same thing because they're just homolog. So it doesn't really matter how you write it. The network is breaking. No, the network is breaking. Do you want to repeat yourself, please?
I said, can we have ataxin 7 over
if, right? You mean ataxin 7 over if?
Yes, can you hear me?
Yeah, I can hear you now. Okay, yes, that's correct. So yeah, so ataxin 7 over if is your question. Yeah, you can have it, but it doesn't really matter, like I said. So either you have curly over if or Y over W, it doesn't really matter. Makes sense. So you can write it the way you want, in as much as you're writing the right genotype. So the orientation on the chromosome, whether you put the top, like something on up or down, or does that make sense? Doesn't really matter.
Okay, so I understand the crossing, but I don't really understand how to differentiate the male and the female, like the W over plus, W over Y. That's where I have issues.
Oh, very good. Okay, so here, so because we know that nothing like there's some things on the Y, but we know that for now, let's assume that nothing is on the Y. So we know that on the first chromosome, Y is gonna, so if you've got plus over plus, you're referring to 2X chromosome. Is that okay? So for male, it's gonna be plus over Y. Does that make sense? And you're just gonna inherit that. You're gonna inherit Y as if you're inheriting X as well. So it's just the same form of inheritance. So for these now, so here now, W over plus, we will know that that is gonna be a female because that hasn't got Y. Makes sense? And here we know that this is also gonna be female. We know that this is going to be male because it has got Y. So assuming you're like plus over plus. So plus over plus is as if you're crossing female and female, and that won't give you anything. So you need plus over Y, which is a male, and cross to female to give you something. And the reason why we're setting up the cross this way around, so assuming you use plus over plus female and then W over Y, you won't be able to dilute your plus because we want to dilute the plus, right? So that's why we use male.
Okay.
Does that make sense? So, but here, so, and then, you know, now we've been able to dilute it out and then we've got our W over Y. And the reason why we're using W over Y, if over ataxin, we're going to get that clearer in the next cross. Does that make sense? So because we want to generate a stock that has got ataxin over Kelly. So, and brilliant suggestion here that we've got other things that we also, other offspring as well. So W over plus ataxin over Kelly. So we don't need that as well. Is that okay? But they're going to be part, you're going to see them because that is going to be viable. And also you're going to see these males as well because they're going to be viable. But you can tell all these apart because these will have curly, but they won't have the irregular facets. So the if balancer, the if marker, they're not going to have the if marker. And these will have both if and curly. This will have curly. This will have curly as well. Makes sense? So then you can tell that, okay, this is what I'm looking for. Yeah, Chizoba.
Sorry, the one you circled, W over Y, if over that, is it because we need the male?
Yeah, you mean
is it because we are interested in the male we are taking that?
Yeah.
The one you circled.
Yeah, we are interested in the male, yeah.
For this, okay.
Exactly, that's correct, yeah. We're interested in the male. Right.
Okay, so I have a question.
Please go on.
Okay, at what point is the genetic crossing coming first? Is it before we cross them, we cross the flies with their drivers, or after we cross with the drivers?
Very good. Very good question. So this type of cross is, so you set up crosses before you do the experiment. So when you cross to a driver, it shows that you want to do an experiment. Is that okay? So crossing or a very big crossing scheme is just, okay, you know what? I need this driver on let's say let's say the driver is on, let's say it's on the third chromosome, just to make things easy. So it is on the third chromosome and you want to look at the poopy. Is that okay? And then you know that, oh, I won't be able to tell if they've got a driver or not if my balancer is MKRS, but you want to use a balancer called TM6B because that can tell you poopy because they're going to be short poopy, right? So then you can be like, okay, I'm gonna move my GAL4 onto TM6B. So you can do a crossing scheme to generate that. But if you don't need that, you can just cross your GAL4 to your UAS and then that's your experiment. So then you can test your drug, you can test if they die, you can test other things, the phenotype, and that's your cross. And actually I've got another cross that my second and last cross that would tell us about that, and then we can ask questions about that as well. Make sense? Right. So yeah, so here we are interested in the male, and yeah, and here the reason why we mentioned earlier on that why are we setting up the cross? Why do we need a cross? So I've mentioned some of the reasons that maybe you want to do some recombination, and that's one of the reasons for this particular one. So every cross has got its own objective. You have to have a reason to do the cross. So, but here we need male. Is that okay? So I'm just going to draw an arrow there. So we're going to pick our male. Is that okay? And then cross that to our stock, which is that W over W. Is that okay? And then now you can then make a stock from this. So just similar to what we've done earlier on, you're going to have several genotype from this, just with a permutation and combination, you're going to have several genotype. But the genotype that we are actually interested in is W over Y. Okay. Over Kelly. So, and I need to mention that the reason why we picked male here in the first place was that if is a marker. So if you pick a female, then actually that won't help the cross because there can be recombination, and you don't want recombination in your cross. That's one thing, and that's the reason why you need to when you're using markers, try and pick males. But when you're looking at balancer, for example, here we're looking at balancer, so you can use female here, but we won't use female because we want to dilate the first chromosome. So you have to look at the reason why should I pick this? Why won't I pick this? So looking at the positive, you have to look at the negative as well. And for these, so we cross this male to our, so we call this a stock, so stock, balancer stock makes sense because it has only got Eve, which is a marker, and then Kelly, which is a balancer. And then you want to generate a stock from them. So just similar to the permutation and combination, you're going to have as part of your offspring, you're going to have W over Y. Is that okay? Which is a male, and that's going to have ataxin over Kelly. And you can tell because you can only have ataxin over Kelly or ataxin over Eve. Is that okay? In this particular cross. And then you can tell that, okay, anything that has got just Kelly is what I'm looking for. Because if you've got ataxin over Eve, that won't have Kelly, it's going to be straight wing. Is that okay? And then it's going to be rough eye. Eve over Kelly is going to be rough eye. And then Kelly as well. So you're only looking for one visible marker, which is Kelly, and then that's what you're going to pick. And then the same thing goes for ataxin 7 that is female as well. So you're only looking for female that have got Kelly. Is that okay? And then another thing is that they are going to be white-eyed. Is that okay? So you can tell that, okay, yeah, this is what we're looking for because they're going to be white-eyed. And also here in this, actually, we should mention that in this cross here, these are going to be red-eyed. If they survive, they won't anyway, but these are going to be red-eyed because this is W over plus. Is that okay? So because of the plus is going to override W, and then it's going to be red-eyed. But here you've generated a stock. So now you need to cross these together, interse, and then you can generate your stock. And then that is what you're going to need to do downstream experiment. For example, you want to do recombination. Can we combine with other stuff? So, or you can actually use that for it. You can use it for experiments. So you can just use it as it is. So, and then, when you want to combine with other stuff, you need to design another crossing scheme for that. Makes sense. But, the beauty about crossing scheme is that, you need to ask questions about it. And, sometimes you forget that, there should be another string that I should have, for example, the W over plus that we forgot. So that's something that you can ask questions about, and people will put you through as well. So yeah, so questioning about crossing scheme is always very important, because once you get it right, then you're good to go. Because if you, like, for example, if you're thinking that, you've got attacks in 7, but you don't, and then probably you've got something else here, and then you publish something about it, it's like, unknowingly that is fraud, because you're publishing something that you're not really working on. Makes sense. So, and then, that will lead to retraction of the paper, because you're showing something that, you're not showing the right thing. So, and that is unknowingly, it's not your fault. Makes sense. So that's why it's really good to be careful when you're designing the crossing schemes as well. Makes sense. Right. So, yeah, so let's go to, so this is the last slide, and I think it's the last cross as well. So, right. So, This is related to, so I think we sent you some slides sometimes ago as well, I think for the workshop, and I think Ross talked about this yesterday in his talk. So this is just a bit of a straightforward cross and a direct cross. Is that okay? And actually, like you mentioned earlier on, that, assuming we've got, so assuming we've got, right, so assuming we've got a GAL4, and then we've got another construct, so we can easily cross that, and then we don't even need to care about any crossing scheme or anything. So, but like I said, you might want to have other things. So, for example, the curly that you've got here is really an adult, is really adult marker. So it's going to mark just adult. You won't see if larvae has got curly or not. You won't see if puppy has got curly or not. Is that okay? So, but there was a balancer now that we've got for second chromosome, actually. So there are two balancers now. So one is called SM5TM6B, and the other one is called CTR. So let's just use the short form. So I'm going to write that later. So we've got CTR. Is that okay? So you can design a cross that, I want to move GMR GAL4 over CTR. Is that okay? And then I want to be able to cross that to this, my lovely US construct. And then I want to be able to tell if I've got puppy that puppy that are going to carry my GAL4 and this, or puppy that will lack it. So, and not only are you going to put your GMR GAL4 over curly, but also you can put this over CTR or SM5TM6B, sorry. You can also put this over that as well, so that you can track, and then you can see your inheritance. Because the beauty of these balances is that you want to be able to track and see your inheritance. Is that okay? And also remember that although these I wrote curly here, but sometimes they can be homozygous variable. So assuming it can be GMR GAL4 over GMR GAL4. So that is not a problem. You can lose the curly if the flies are happy with the two with homozygous form of this GAL4, if that makes sense. And the same thing goes for the UAS construct as well. So if they are happy with the homozygous form of it, you're okay. Is that okay? So you don't really need to cross it to CTR or anything. But assuming you're like, I want an internal control. So you can use, actually CTR is a very good internal control for your cross as well. And, I should mention that, hopefully, I didn't forget to mention that later on as well. So yeah, so what you can do, use this type of experiment, actually. So I gave a preamble that you want to test if a kinase called RET. So this is a kinase. Is that okay? And actually is a mutation in this kinase that we're really interested in. And the mutation is mutation from methionine to T. Is that threonine? Yeah, that's threonine, isn't it? So yeah, so to threonine, is that okay? So then you want to look at the effect of this mutation. Is that okay? And then you want to look at the effect of this mutation in the fly eye. So, you're beginning to answer and ask your question, okay, the fly eye. So what do I need? I need a GAL4 that is expressed in the fly. in the I mean. Is that okay? And then you have got these two strings. Is that okay? So, and what GAL4 expresses in the I is GMR GAL4. So because, you know, so these were identified and discovered just, you know, people were doing this and realized, oh, you know, these actually are from, GMR is probably like a gene. Is that okay? And then it has just got GAL4 attached to it, because GMR, people probably stain, you know, antibody stain, and realize that, you know, there's a localization of GMR in the eye. So let's tag some GAL4 to it, and then we can express some things via that construct. So that's the way GAL4 are being discovered. Is that okay? And, you know, it's just, that's why it's sometimes cell type specific, you know, the particular gene that is expressed in this particular cell, you can have a GAL4 for it, and then you can express gene to that particular cell type. Makes sense. So that is, so assuming you've got a gene that only expresses in the wing, you can have a wing GAL4, and then, you know, you just give it a name, and then that is, we're good to go. So then you can use that to express your US construct in the wing. But for our own case, we want to express that in the eye. And then you can begin to be like, what is the genotype that I'm going to look at? And then what is the crossing scheme that I'm going to be interested in, or how will I design my crossing scheme? Is that okay? And at the end of the day, this type of experiment is, you can then, one thing you can do with this experiment that has been shown in the lab in the past actually is, when you cross this together, gives rough eye phenotype. So the fly is going to give a rough eye phenotype, or the eye is going to be rough. And then the next thing you can ask is, you know, can we treat this? What are we going to drop on this to cure it? That's one thing. Another thing is, you can begin to look out for genes that can modify that activity. So, oh, we've got rough eye. Can we have a gene that makes it worse? Can we have a gene that makes it better? So these are the questions you can, you know, you can ask with this type of experiment as well. And I would love to tell you that, you know, that is just a thyroid cancer model. So you've just designed a thyroid cancer model. So yeah, so that is really, so that is how things are being done, and you know, that's why you need crosses. So yeah, so let's go to that cross now. So if I go back, so you can take a picture of this just to remember what the genotype looks like. And then we can look at the cross, and then we can wrap it up from there, I think. So right, are we good? Good. So right, so I'm going to change that to, so our number one stock, we've got, so I'm just going to write, and then our number two stock. So I'm just going to write the first and second. So because the first actually doesn't really matter as well, but it's okay for this. So then we've got our UAS. I'm just going to write direct. Is that okay? So we know that is a mutation. Is that okay? Then you, like, the first thing is what, so the first question A was, what is that genotype that you want, that you're interested in? So your genotype is just, you want GMR. Let's say here we're looking at adults. You want GMR GAL4 over UAS direct. Is that okay? So that is the genotype that you're interested in. Is that okay? And like I said, this, how would you generate this? So B, what you just need to do is here, because your GMR GAL4 and your US rate, they're both on the second chromosome. So you don't need to worry about using male or female. Here, you can use either male, you can use any. Is that okay? So just for the sake of time or argument, I'm just going to use male from GMR GAL4. So I'm skipping the plus. So this is the same thing, please. So, and then UAS, actually, let's put the plus there. Plus over Y, semicolon, plus over plus, semicolon. And then we've got direct over curly. Makes sense. Now we're going to have several offspring from that. So one, we're going to have, start from here, plus over y. Is that okay? Plus over y, plus, so plus from there, okay, y from here, plus from there. And then we're going to have GMR from here, and let's say Kelly from there. So, or even let's play with this first. So then we can have GMR gal4 over USD rate. So let's play with what we want first. So GMR gal4. So you can honestly, it can mean any other. I'm just playing with it. Is that okay? And or we can have plus over plus GMR gal4. And it can also be USD rate over GMR gal4, so it doesn't really matter. So you're just writing it. That's not gonna, like the genome is gonna do what it's gonna do. So, or you can have plus over Y, GMR gal4 over Kelly, or plus over plus GMR gal4 over Kelly, or plus over Y UASD rate over Kelly, or plus over plus UASD rate over Kelly. Right, so here we only need this and this. Is that okay? So, and I would say that please don't ever use this like this from this, because one thing is just bin them. One thing is, we might be tempted to be like, GMR gal4 over Kelly, I can still use it. I can use that in place of this, but actually it's different, because one thing is you won't be able to tell, though you can write this mathematically, this is the theory, we can write this, but actually in the real world, you won't be able to tell if you're inheriting GMR gal4 or USD rate. Is that okay? Especially because they have got the same I column. Is that okay? And you've got Kelly, so there won't be any way to to decipher if they're different or not. So you just need to look at something that hasn't got any balancer. So, and the feasible phenotype here is red-eyed, no balancer, red-eyed, no balancer. Same here, balancer is crossed out. I don't need them. Makes sense. So these are the flies that you're gonna look at. Is that okay? And another thing is, you cannot generate a stock from this. So you can't cross these two together and be like, I want to generate a cross. No. So that was why I said there's an experiment and there is a cross. There's a crossing scheme or a cross that you need to design. So here we're not designing a cross, we're doing an experiment. So, and you've done the experiment that, you want to look at the effect of DRTC using a GMR gal4, and the effect is the only thing you need to look at in this offspring. This offspring, after that defect, they're useless. You can't use them for any other thing. Makes sense? So, and anything that comes out of the cross, because it's an experiment, you have to bin it. And if you want, then you can design. So there are, you can then, assume you want to do several other things, for example, like moving things around. Like I said, assume you want to move this over what we call CTR. So CTR equals Kelly, Toby, and RFP. So you can move this over these. You need to design set of cross for that. I think you want to move it over something called SM5 TM6B, because these are balancers and they've got balancers and markers for second chromosome. So then, you can move it. And actually this is a co-joint balancer. So you can use this to balance the second and third chromosome. But this is a very good and actually fabulous balancer for for the second chromosome. And like I said, so the phenotype here, you can have Toby, you can have red, you can have RFP, you can have Kelly, actually three markers. And here you have Kelly and Toby. Is that okay? So that is the marker. And Toby is very good because you can look at larvae, you can look at pupae, but Kelly is only for adults. So, you know, you just have to look out for, oh, what do I need? And you do the same similar stuff, you know, with the first cross that we did, you know, we move a bit of things around. You just design similar cross. Oh, I want to move things around. What do I need? What type of stock have I got? And, you know, what stock do I need to order? Who do I need to get it from? So these are questions that I know that you need to bear in mind when you're designing your cross. Is that okay? So what do I have in my lab? Things like that. So these are the questions. So yeah, right. So any question based on this, please? Based on this particular cross. Right. So I take that as a no. So right. So that is, yeah. So that is it for the crosses. And, you know, like I said, we can always go back and, you know, talk about them, and also, you know, go to like complex ones as well. So there are a lot of complex crosses out there that you can always play around with, and it's really, really fun to do. So
I have a question.
Yeah, please do.
But this is not about the crosses, the genetic crosses. It's about the maintenance of this particular type of flies.
Okay.
Okay, so because we have something like this is GMR, yeah, and compared to other type of flies, it seems to be doing too well in the other way around. So I don't know if there is any special care that we need to put in place or something, because we have tried everything and it doesn't seem as if to be they're doing too well. Something like that.
Okay, so you mean, so the stocks. Oh, so what you can do is, so to maintain the stocks, what I recommend is, one, make sure that you probably grow them as, let's say, 25 degrees or 25, 22, like just around that room temperature. Is that okay? That's one thing. And secondly, you need to, you can also flip them like every 3 days, you know, flip them into new vials like every 3 or 4 days. Is that okay? So because when you flip them maybe every day, you know, they might not lay as much egg as possible to really match up the food and make sure that the larvae can crawl out. So, and sometimes it is just that, I don't know if I remember, I've forgotten what we were saying, but yeah, if that was what we sent, but yeah, maybe that was SM5TM6B, but I think this is what we sent. So, but yeah, so I would flip them like, you know, every 3 or 4 days and make sure that they're grown at, you know, at room temperature. That would be the solution to that. But sometimes, you know, maybe mite infestation as well can cause it, but like can cause them to be sick. But you just have to really look out for them carefully. Another thing is, you know, once you've got like flies coming out, you know, make sure that you don't have too many male over female. So assuming you've got like, let's say some virgins coming out, you can have like, you can pick about 5 of them and cross it to like 2 male. So 5 female to 2 male. And that also helps the cross a lot. Is that okay?
Okay, sir.
Yeah, so that also helps the cross a lot. So, but yeah, but sometimes some crosses, especially if they've got a lot of balance in them, some stocks are always really sick. You just have to look out for them, look out for the temperature that they're very happy in, and then you're good to go. Right.
Yeah. Please. Yeah, you mentioned something, temperature.
Yeah.
Yeah, this particular fly she's talking about, DGMR, it came with the other one, I think wheat, right? This wheat. Yeah, it came. Yeah. And that one is doing pretty well. So does it mean temperature dependent for the 2 of them? They came at the same time. The other one is doing very well. Oh, now I talk about temperature.
Oh no, I see what you mean. No, I know. Also very good. I like that. So you're absolutely right. So the temperature that I was referring to is sometimes some stock are just like that. So like I said, you know, like D-Red is just happy and then it's doing his own thing. But GMR Garfor, I don't know what would be wrong with it, but because I think it was doing very well. Because I think I was the one that sent that to you actually. So I think it was doing very well in the lab. I can check again, but
okay, we have 18-degree incubator. Do you advise we put inside the incubator to check?
Degree
to maintain. I think it's 18.
Yeah, so you know what? Stock like that, I won't put them at 18. So I would throw them at room temperature, like, you know, like 25, if you've got 25, even if it is 27, I don't know, I don't remember the temperature of Ibadan now, but, you know, if it is 25, if it's 26, you know, a bit of high temperature will really allow them to grow faster and allow them to really be happy. So, and like I said, you know, don't change them probably every day, but try to change them maybe every 4 days or 3 days, and then that allows them to mate properly. And also don't have too many male in the, you know, in the vial.
In one vial.
Exactly. So make sure the female are more than the male. So yeah, it happens. So not necessarily because you receive them at the same time, but you know, it's just the stock that is playing around and just messing around with us. But yeah, it's yeah, no problem at all. Yeah, we've got people raising their hands. So please, you can just, you know, you can interrupt, please, because I can't see the names. That's a problem.
Yeah, sorry, I wanted to ask concerning this.
Yeah, the network is breaking, unfortunately.
By the time we cross them, what are we looking for? Are we looking for Corley and Toby to show that they are cancerous? Can you hear me, please?
Yeah, I missed that question. Very, very important. So please,
can you repeat it?
Yeah, I can hear you.
Okay. I will take it again. Do you see GM Carol for and the USB red flies? By the time we cross them, what are we actually looking for to show that they are cancerous? Like they produce cancer. Is it coily turbid that we are looking for? I understand the way to cross them, but what are we going to look at for? I know both of them, but how do we phenotypically, what do we see to show that these are cancerous flies?
Very, very good point. So like I mentioned earlier on that, you know, the GMR-GAL4, you're looking, should be in the eye. So, and sometimes, okay, I would mention that sometimes it can be lethal. So you like, you know, it can kill the animal, but those animals that survive should have rough eye. Is that okay? And that's a phenotype that you're looking for, and you know that has been published in the past as well. So you know, so look out for rough eye phenotype. You know, they've got the normal eye, and then you're gonna see this rough eye that I think Ross might have mentioned yesterday as well. So that is the phenotype that you're looking out for. Is that okay? So
thank you.
I think it shouldn't be lethal. I think you should see rough eye. Yeah. Yeah, if it is not lethal, then definitely your phenotype is rough eye. But if it is lethal, do let us know, because I mean, like, if nothing survives, you know, if what you always generate is curly, because you don't need the curly, right? You only need, you know, straight wings, like so, and the straight wings should have rough eye. So that is the phenotype that you're looking for. Right. Any other questions?
Sorry.
No problem at all, please.
Thank you so far for the presentation. It's been enlightening.
Thank you.
And to what I just wanted to ask is in relation to the question that was formerly asked on how to maintain the flies. You could help us with your food, your diet composition. I think that may help a little to know what they are being fed with there to make them survive compared to what we are doing in Nigeria that is making them not survive. I'm currently out of Nigeria. I am doing my doctorate in Brazil, but I can remember there was a time when we got some flies and they were not surviving. We had to change the composition of the diet. We added sucrose, we added milk, and they were surviving for a while, but then I think they got used to it again and started dying. So maybe the composition of your meal there can help, I think.
Yeah, no problem at all. Actually, that's really right. That's actually a very good point. But the thing is, like most labs like ours as well, I think the composition of the media is the same, like it's similar across the world, because and people use so Bloomington, I think they've got a recipe for for fly food and things like that. But in when I was in Liverpool, I do remember that it has got like sucrose, it has got yeast powder. So yeast powder is actually really good. So even if you add it, as you've got your own food and you just add yeast powder to it, like flies lay eggs a lot when they're on yeast powder. So, and it has got corn powder as well, and it has got one last thing. Yeah. So it has got these 3 or 4 things, that allows them to grow properly. But I'm going to check with Glasgow and see what is being used in the lab here. But
sorry, is it the preservative? Would they affect it?
No, I don't think.
I'm not sure, because I don't think you need. I don't know if you need to add preservative, because it depends. Are you making them yourself or you're buying them?
No, we are making it. We are using Nipagine, methyl
paraben as the preservative.
Yeah.
Oh, I see. So yeah. So So because I see what you mean. So yeah, I think you should reduce. Yeah, sometimes that kills flies as well. So you should reduce the concentration. That's correct. Yeah. So because that's like a preservative, is an antifungal and antibacterial. Yeah. So yeah, so you should reduce it. So that's why sometimes we use propionic acid as well. But just make sure that it's not too much. And when you can make like not too many batches of food and just a bit, and then you can grow them on it and see
for them. Okay, like we should change specific for that particular file for now.
Exactly.
Around now we're using 0.5 gram for about 52 gram corn meal, so we can reduce it.
Yeah.
Judith, I think you should calculate the concentration of the Nipagine based on the diet that you're preparing. Yes. Calculate the concentration, maybe 1 millimolar or 1 micromolar. Then I think you should just calculate the concentration, make like 4 different concentrations and work with it, and let's see the effect afterwards.
Okay.
Yes. So I think you can be able to write it down that, 1 micromolar for 100 ml of water, or 1 millimolar, something like that. Just make like 4 concentrations and let's see the effect on the fly afterwards.
Yeah, absolutely. No, absolutely. That's a very, very brilliant idea.
Thank you very much. So we'll take the best from there.
Yeah, and like I said, the yeast as well. So I think like normal yeast for bread is gonna do the job. So just add that to the vial even after making the food. It's really
okay. So we can sprinkle, Judith. We can sprinkle yeast. We sprinkle yeast on the meal after cooking with yeast.
Okay.
You can have like dry yeast on the food.
Okay, dry yeast on surface of the diet.
Yes, on the surface of the diet after cooking.
You can put it at the center.
Yes, sir. Yes.
Or you mix it with your solution, make it into a paste, and then put it at the middle of the diet.
Yeah.
Okay, sir. But are we still going to use? We are still going to use the yeast when preparing, because
we are still going to cook with the yeast. Yes. We are still going to cook with the yeast, but then we still sprinkle a little quantity on the surface of the food. Yes.
Okay, like uncooked yeast now.
Uncooked yeast. Yes.
Okay.
Yeah, that's correct.
Thank you.
No problem at all. Those are fantastic questions.
I'm very sorry, Dr. Amos. I asked many questions. I'm sorry.
No, please do. That's the beauty of the history. Please to ask a lot of questions. Hello, please.
Hello, sir. This is Jafar. Sorry, can I have just a very simple question? Thank you so much.
Please go ahead.
I actually wanted to know if there is different
That is breaking. I'm so sorry. That is breaking. Do you want to repeat yourself, please?
Yeah, I said, sorry, I wanted to find out if there is a difference between ELAV GAL4 and then the GAL4 alone as a driver.
Oh, very good. So GAL4 alone is never going to do anything. So GAL4, so if you're like, I inserted GAL4 into the system, no, it's never going to do anything. So it has to be tagged to a promoter. Is that okay? And the promoter can be a promoter of, so that is just that is a location. We can just be like, it's a location. Is that okay? So an ELAF is a gene, that is expressed in the neuron and things like that. and it is pan-neuronal. And you can be like, okay, when I tag my GAL4 to ELAV, I should see expression of, let's say, GFP in neurons. Makes sense. So, and it's just similar to GMR GAL4. GMR is just you can just replace GMR with ELAV. Makes sense. So you can have GAL4 on its own, but you can have ELAV GAL4. You can have anything GAL4 these days. Makes sense. You can have exokinase GAL4, and that's going to tell you where the location of exokinase in the system. So you can have actually virtually everything GAL4. So you need to tag a particular promoter. It could be any gene that you're interested in, and then tag that to the GAL4, and then you can use that to drive things in the location that you're interested in as well. So that is there.
Right. No problem. Okay, I want to add something to the ELAV. Oh, can you hear me?
Yeah, I can hear you. Please go ahead.
There was a time I was also making inquiry. So I found out that the ELAV means, I think, embryonic leader abnormal visual system gene. They said it's specific. That is the full meaning of the ELAV. You can check it online.
No, that's absolutely correct.
Yeah, it's specific for the neurons. They say it's important for formation and maintenance of nervous system in the fly. So that means most times the expression when you use ELAV GAL4, most time we look out for it is for neuron-specific protein. So we look at for the eye. What I was able to find out when I checked on Google.
Yeah, no, absolutely, you're right. No, absolutely, that was really fabulous, and you're absolutely right. So, and you could express DRET with ELAV and see, there are several things you could do. Just try them out and see. You could express it with ELAV and see what will happen. What will happen to the neurons, or or to the motion or the locomotion of the organism. You can, yeah, absolutely, you're right. You're absolutely right. Well, I think we've got questions.
Thank you.
Yeah, no problem. I think there's someone raised their hand. Yeah, please go ahead, please.
Okay, thank you for the beautiful presentation. I want to know if, like, going through a journal now and see like a motor that you're interested in, what could prompt your interest? Which one could be the male? Which one could be the female? Or anyone can be the male or the female? So I don't know if you have answered this earlier. I have network issue.
Oh, please, do you want, I'm
That's
so sorry.
Question.
It's actually I missed the important point. Do you want to repeat the question?
Okay, the question is that before you order for a fly, can you actually dictate which one should be which gene should the male be? On which sex should your gene of interest be? Is it really important? Like, should it be on the male or on the female? Which one should actually carry the gene of interest?
That's a very good question. So you don't, so beforehand, because you can have your stock without male and female. So that is one key thing. Does that make sense? So your stock will always come with male and female. So for example, here, the GMR over Kelly is a stock. You will have male and female there. The US DRET is a stock. You will have male and female there. Makes sense? But in your cross is dependent on what you're looking for. Is that okay? So let's say, for example, earlier on that we were designing the cross, that we're looking at the crossing schemes or the attack scene. We know that if we use male, we won't get what we're looking for. So we decided to use female. So you have to look at, okay, what am I looking for? Am I looking to make something more white or something more red? So, and we know that white is on the first chromosome, right? So it actually really, when you're looking at something on the first chromosome, that's really where male or female really comes in really important. Is that okay? And also when you're looking at maybe balancer or marker. So because you want to use male when you're using markers, and you can use female when you're using balancers. So those are where the question lies. So you're going to order any fly that you want anyway, but when you're designing a crossing scheme that involves you combining things over time, then you have to be careful of, do I want to use male or female? And that's dependent on what you want to combine. But when you're setting up an experiment, something like this, does that make sense? Like we said, we don't really care here whether we use male or female. It's only, so actually the rate limiting step here is what virgin have I got? because you need virgin, right? So have I got more, like, for example, the people were saying that, oh, DRET is actually doing very well in DRTC, then, you know, I would just do DRET female. And because I only need maybe 2 male for 6 female, you know, I'm just going to use GMR male. You know, that would be the solution to that experiment. Does that make sense? But when you're looking across over time, you know, then you have to be careful that, oh, do I want to use male or do I want to use female? Why do I need to use male? Why do I? So you have to ask. So it's about why, you know, and it's about how. So why and how am I going to do it? So that's really the question. Makes sense? Any other question, please?
Yes, please. I have a question.
Please go ahead.
Thank God Miss D is online, so she can also answer.
No
like, okay, now I love there's a particular work we are doing. We are crossing pink 1 and ELAB. Why are we using the male pink and the female ELAB? Thank God Miss D is online.
Right. So I think is it UAS pink 1 or?
Yeah, UAS pink 1 and ELAB gap 4. This one we're using the female ELAB, the male pink.
Very good. So, and do you know if ELAF is on the first or second?
I don't know.
I think it's on the second. I think ELAF GAL4 is on the second. But
yes, it's on the second chromosome. It's on the second.
Okay.
So I think, you know, if you do it the other way around, it doesn't matter. Makes sense. So the reason why you could be using it is because, like I said, you know, one of them is doing greater than the other. You know, happy fly, you can collect lots of virgins because, you know, you need to time virgin, you need to make sure that you've got enough of them to set up cross. But actually you can start a cross with 2 male. Sometimes I start cross with 1 male. Like, it's just depending on what I want to do and how many or like how large I want to go. Does that make sense? So for this type of cross, like I said, you know, whether you use male or female, it doesn't really matter because they're on the second chromosome. The same thing goes for ELAF and pink 1 as well, because, you know, both of them are on second chromosome.
Yeah, pink 1 is second chromosome. Yeah, I think it's pink 1 over cryo. Yeah, second chromosome.
Makes sense.
Thank you very much.
It won't really matter. So like I said, the limiting factors to use male and female is, you know, do you want to track something, and are you on a balancer or are you on a marker? So is my gene on a balancer or is it on a marker? So if it's on a marker, then I want to use male because recombination will take place if I use female. And do I want to dilute the plus on the first chromosome, or do I want to dilute the white on the first chromosome? So, you know, these are the things. It maybe not white, because in some crosses that we've done in the past, we've got like Hitchcock flipase on the first, and then we want to combine Hitchcock flipase with something. So we know that, Hitchcock flipase, we might need to use female of that because we want to make, we don't want to dilute it. We want to make sure that this new flies has got it. Does that make sense? So, and the same thing, we've got eyeless flipase. You know, you've got other things on the first chromosome that, you know, that you can, now actually you can either dilute out or you can add to things. Is that okay? So, but if you've got like second chromosome, you can use any. So it doesn't really matter at all. Right, Olushola. Yeah, I can see.
Thank you very much, sir, for the nice and wonderful presentation. I think
thank you so much.
As I am now, I think you have widened my knowledge in the area of this research. Please, I just want a clarification on something because I lost contact. I think with the presentation at a point at the first slide on the nomenclature. Please, I just want to clarify something there.
Okay.
Just let me see the first slide on the nomenclature.
No problem. We're going there now.
Thank you very much, sir.
Right, first slide.
First one on
the
nomenclature. No.
Right.
Oh, the first. Yeah, this one. Yes. Okay.
Sorry about the writing.
Yeah, I understand. Okay. I think because in these the absence of homologous. Okay, the TM6B. Okay, at the last part, I didn't really get what you were writing there because I wasn't part of this thing. My network failed at that particular time. So I just want to know what they stand for so that I will be able to.
No problem. Thank you so much for the question. And I'm very happy that you really brought this up. Thank you so much. Right. So this particular genotype that I wrote here, so this one, so Y and W over Y and W. So this is homozygous. Is that okay? So
yes. I got that.
Makes sense, right? So, but I'm saying that, you know, you can also have homozygous, but not necessarily write it twice. So in the Drosophila world, assuming you write US MIC, people will assume that it is homozygous. Just US MIC on your second chromosome. Makes sense. So people will assume that, oh, that is homozygous, that's okay. So, and then here, then you can have, and so these are actually balancers on the third chromosome.
Like the CYO2.
Exactly.
CYO is,
on the second. So the CYO is on the second, but TM6B is a balancer on the third. MKRS is also another balancer on the third. And actually, that someone asked a question earlier on that, oh, if I see like a balancer, does that mean that I need to look there for my gene? Actually, because someone like me is working with MIC, I will look, I don't care about this TM6B or MKRS. I only care about MIC. But actually, no balancer is on MIC. Does that make sense? So having the balancer there doesn't really matter, it doesn't really make any difference to this talk. It's just to look at what you're interested in.
Makes sense? Thank you very much, sir.
No problem. Thank you so much for the question.
You're welcome, sir. Yes, sir.
Hello, sir. Good afternoon, sir.
Afternoon, sir. Thank you.
Now, as regards the crossing earlier stated, using of PINK1, UAS PINK1 and ELAVI-GAL4.
Okay.
Now, we know that from evolution theory that there's no recombination in the male fly, for instance, in the second chromosome. That's why it's advisable to use female ELAVI-GAL4 and male because in the male actually there's no recombination. So to have a specific gene of interest, it's advisable to use the male PINK to cross with the female GAL4.
Oh, I see what you mean. So when I say, so when you're driving things, you're not driving things through recombination. Does that make sense? So when you drive things with GAL4, it is not through recombination. You're driving things because you're expressing two things. And because they're in the genome, they're going to bind. So this is an interaction. It has nothing to do with the chromosome. So you've got this chromosome that has got this thing, and then the homolog has got that. In the genome, we've got a transcription factor and our gene of interest or our UAS. So GAL4 is going to walk and bind to UAS. Is that okay? And then UAS is going to activate the expression. So because it's an upstream activating sequence, so it's going to activate the expression of your gene of interest. So as if you're looking at GFP, you're going to say green. As not necessarily green, but you're going to see the fluorescence. Is that okay? And then, assuming you're looking at RFP, you're going to see red. So, or you're looking at a particular gene, you're going to see your gene. So it is actually like a biochemical reaction going on there, but in all organism, rather than in our test tube, if that makes sense. So it is. So your expression is not via recombination. Exactly. Recombination is something different. So you can recombine things without expressing anything. Is that okay? So that is why it doesn't really matter whether you use male or female when you're setting up some cross. If you're got something on second chromosome, like for example here we can cross this to ELAV-GAL4 because this is US MIC. So anywhere you see UAS, it's mostly people will cross it to GAL4. So it now depend on the type of GAL4 that you want to cross it to. And like he said, is it ELAV? Am I interested in neuron? That's your question. What phenotype should I look out for? Those are your questions. Makes sense. So, and yes, the crosses. So the crosses is only dependent on, your interest, whether they're on second or third, or whether they're on first and second, or whether on first. So if assuming ELAV is on first, now let's go back to that. So assuming ELAV is on first and PINK is on second, Is that okay? So if ELAF is on first, I would definitely use female, because you know that all your offspring is going to have one copy of ELAF. But if it is male and then you use ELAF male to cross to pink, then it is only the female at the end that will have ELAF. Male will lack it because it is ELAF over Y. Is that okay? So an ELAF over Y is going to always go to ELAF over plus. And then at the end of the day, you're going to have plus over Y. So which would have ELAF. Does that make sense?
Yes, sir. Yes, sir.
Yeah, thank you so much.
Thank you, sir.
That was really a fabulous question. Thank you so much. Thank you so much for that. Wow, I'm having this block noise. Right, so any other question, please? Yeah, those are really, really beautiful questions. Thank you so much for that. So, right, so I think, yeah, I'm almost in the last slide. So, right, so I just want to show you some, some online resource, like I said. So Flybase is awesome. They've got different.
Hello?
Hello?
Can you hear me?
Yeah.
Sorry, sir. While you are answering the last question, actually my network had issue. I didn't actually listen to the answer you gave. Can you take yourself a little bit back?
Yeah, absolutely. That's fine. So yeah, what I was saying was that, so actually if we go to the cross that we set up, so I said, assuming your GAL4 is on second, and the gene and your UAS construct is on second as well. So you don't need to you can use either male or female, because the expression of these genes in that particular context is not dependent on recombination. Is that okay? So because whether you have male or female, you will still express your thing. Because, I mean female, or whether you have male or female offspring. So for example, actually, if we go to this one. So for example, here, male or female, they will still express the thing that we're looking for. Makes sense. So it doesn't really matter whether we use male or female for starter. But assuming GMR GAL4 is on the first chromosome, actually this slide is very important as well. So it's on the first chromosome, and UAS is on, and US direct is on the second. Is that okay? Then you cannot use male, because if you use male, then GMR over plus is the only thing you're going to get. You won't be able to get GMR over Y. Make sense? So yes, you won't use male in that context. But assuming you use female GMR over, because female GMR, that's going to be GMR over GMR, because plus over plus is on the first chromosome. Then you can generate GMR over plus. So let me write that down just to make it better. Right, so here, let's come here. So assuming we've got, so we've got GMR GAL4 over Y. So assuming you cross that to plus over plus, so let's just forget about right now. So because we are interested in chromosome, isn't it? So your offsprings are gonna be GMR over plus or plus over Y. Makes sense. So those are the two major genotypes that you're gonna get. So basically your male is gonna lack GMR, but instead of that, you need to use GMR GAL4 over GMR GAL4, because that's X over X, right? So because it is plus over plus, X over X, and that's the correct way to say. So then cross that to your ret, that is plus over Y. And then in your offspring, you can get GMR GAL4 over plus or GMR GAL4 over Y. Does that make sense? So if your GAL4 is on the first chromosome, never use male. Makes sense? Because you wouldn't get what you're looking for, especially in male. So you can actually, assuming you're looking at adults, you can be like, I know that my male won't get it. So you know, it depends on the game you want to play actually. So, but assuming you want all of them to have what you're looking for, so I would advise you to use female. And you know that, you know, it depends on your compromise and what you need and what you want. And, you know, also you can use the male as internal controller. You know, they don't have the GAL4, so I'm just going to use them anyways. It depends on the type of experiment, you know, the game and things that you want to play. So I would just, you know, but that is where the concern can come in that, you know, oh, why do I need to use male or why do I need to use female? But in this context that we've got them on the second chromosome, you know, you can use either. Makes sense. So, and it doesn't really matter. Does that make sense a bit? Are we good? Just a bit of response.
Yeah.
Okay. Thank you so much. That's awesome. So, right. So, yeah. So we've got some resource. Is that okay? So yeah. So Flybase, I said, is really fabulous. You know, they've got a lot of things on there. So visit the website. I think hopefully they continue to get funded, but because I think there's something going on with the fund these days. But yeah, hopefully things continue like that and get funded. So, but yeah, they've got a lot of resource. You know, they've got the community there. They've got, so Flybook is actually something really good and that, you know, that you can look at. I think that is connected to the genetics journal. So, and also they've got all these bioinformatics too. So, you know, Marvel, gene to function and single cell, CSIC, and, you know, you can look at them and really, they're really good. And, you know, it depends on what you want to use them for, but, you know, they've got all the resources there. And one of my favorite is the interactive fly as well. So you can look at detail of gene. Let's say, for example, you're like, oh, I'm interested in this particular kinase. I'm interested in this particular protein. Can I see the autolog in fly? You know, you can Google the name and then you can be like, okay, the name is this. And then you can actually search this website and then that's going to give you some detail about the gene if it is there. So it's actually really, really, really good resource. And this Drosophila model of human disease is really good by Stephen Moore. So she's really good and she wrote a lot. She did a lot of things with the blog. And, you know, you can also, actually most of the things that, you know, the summary of papers and, you know, tell you about what people have used fly for in human disease, and which is really, really fabulous. She also wrote a book, I think Frt and Flp, really good book as well. So she's really good. And that is by Tony, Thomas Brody. So you can also, he's really a very good guy as well. So yeah, Manchester Fly Facility by Andrews as well. So they've got, I think you guys know about them. So they've got nice resource, Drosophila genetics, you know, they're really good in collaborating and also in teaching as well. So yeah, so very good resource out there. So yeah, Bloomington for you to order flies. Also not just ordering flies, but actually they've got some like, so as you want to look at it, some tools and technology in flies. So they've got, I think they've got a tab for that on the website as well. And actually you can read about this technology and also the application and what you can use them for, and the power is really, really amazing. So these websites are really good. And also the VDRC stock center. So they've got, like I said, inverted repeat and things like that. So you can order these from them as well. So, and yeah, so that has been really, really fascinating. And thank you so much for listening. And thank you so much for your time. And I'm happy to take further questions and also discussion. So I'm gonna stop recording. Is that okay, doctor, for me to stop recording, right?
Yes, it's fine. It's fine.
Good. So